Hi, everyone. Can you hear me? Is this on? Is it working? It's working? Okay. So we're going to go ahead and get started. Hi, I'm Jeannine Cavender-Bares. I'm director of the Harvard University Herbaria. And this is a note to you all that we're supposed to talk into the microphone, otherwise folks online won't be able to hear us. And Jonathan there is going to pass around a microphone for those of you who left any time in your talks for questions at the end. So welcome to the first IHerbSpec Symposium. This is the International Herbarium Spectral Scanning Working Group that many of you in this room have been participating in virtually, and we are so delighted to get to have this in-person meeting. I just want to express huge gratitude to the Harvard University Herbaria and for the National Science Foundation for funding this and bringing us all together. This is funded by iDigBio and also by the Ascend Biology Integration Institute, in addition to Harvard. And thank you all for traveling all this way to be here. It's just a delight. So what we're going to do today is talk about our vision for next generation specimen digitization, or in other words, the international herbarium community goes spectral. And what I'm going to talk about is just an introduction. Many of you are fully aware of these kinds of things, but just to get us all on the same page where we're starting from, what we're talking about. And critically, what we're doing is we're coupling new kinds of biodiversity information with hundreds of years of accumulated knowledge of our living planet in a time of uncertainty in so many ways. So the work we're doing is really, really important. We're safeguarding our knowledge of the past, and we're bringing it together with innovative new ways of understanding and seeing biodiversity. So we have a really tremendous opportunity right now, following this idea of the extended specimen, to bring spectral information into herbaria as a way of getting plant trait data, which some of us have spent our lives working on collecting this plant trait data. It's a really fantastic source of plant trait information, and it's going to take a ton of work to do this, but there's a lot of promise for this. And it's going to be informative for taxon identification. Not a panacea, as Barbara will tell you about, but it's going to really help with that. But to do that, we're going to need to aggregate and work together as a global community so that we're not doing this in 10 different ways or 20 different ways in all the world's herbaria. That's why we're here now, is so that we can figure out how to do these things collectively and agree on protocols and metadata standards so that at the end of the day, we can actually aggregate the data and not go back and have to redo things. So what are we even talking about? Well, let's just start with the electromagnetic spectrum, where we have wavelengths or frequency on the x-axis, and on the y-axis we have energy or reflectance. In this case, it's irradiance, and this is the sun's energy that reaches the Earth, the top of the atmosphere in gray, and the red is what actually makes it to the surface. So the atmosphere is creating all kinds of, absorbing the energy from the sun in various ways that give rise to these peaks. Here's the visible with the rainbow. That's the visible region where your eyes can see. It also happens to be where plants photosynthesize, 400 to 700 nanometers. That's not a coincidence. It coincides with peak irradiance from the sun, and also is kind of a safe energy space. And then when we measure leaf spectra, I mean, we're all doing this in various ways and using different parts of the electromagnetic spectrum, but this is probably the most typical range that we're working with, from 400 to 2500 nanometers. We call this full range, and the y-axis is reflectance. And you can see the difference between a fresh leaf and a dried leaf. The difference is really the water. So spectra from herbaria look different than spectra from fresh leaves. There are features in these spectra this is very high dimensional information. So the features in the spectra tell us about the chemical content, chemistry, the function, the structure of plant tissues. And so we can get a lot of information about plant function from these spectral signatures. So one thing we can do is use plant spectra to detect function. We can also use the spectra themselves as traits or a way to capture phenotypes. And so I've been directing the Ascend Biology Integration Institute for the last 5 years. It's advancing spectral biology and changing environments to understand diversity. And what we've been doing, and Dudu's been part of this and other folks, Dawson, we've been using spectra as a common data type to integrate biological variation, to understand biological processes across scales, from the leaf level and linking that to genomic variation at the canopy level, ecosystem level, landscape level, regional, continental scale level. And using a common data type gives us a way to connect information across scales to scale up. And by spectral biology, we mean what we learn about biological processes when we apply tools that use the interaction of light with biology. So here are two kinds of spectra from fresh leaves, from dried leaves. This is pulled from Sean Kotari's paper, Predicting Functional Traits using partially squared regression analysis to predict traits from the spectra, fresh and pressed leaf spectra. And you can get more traits from fresh leaves, so you get all of these different kinds of traits. But from the dried leaves, you actually get quite a few traits, and often you can predict them better than you can from fresh leaves because you've removed the water that masks some of the chemical variation. And then Dawson, maybe you know, then did a follow-up study to see how well Sean's models worked in actual herbarium specimens, because these are pressed leaves. So there's really good promise that we can predict traits from herbarium specimens and do it well. Why would we want to do that? Well, I'll give you a couple of reasons. But one is there's a whole community out there trying to scale up plant function to the global scale for global models of biosphere function. And they've done this in various ways using trait databases like the TRI database, coupling that with airborne functional trait mapping, and trying to get global maps of plant function and predicting plant traits from satellite data. So there's a whole realm of folks working on that. The space agencies are all involved in this. But they haven't been tapping into herbarium specimens as a source of trait data. So this is something that we could really contribute. And an even cooler thing is, this is a figure from Antonio Guzmán, who's sitting right now. He's not actually going to talk about this, but one thing that can be done to couple specimen plant functional trait data to satellite data is to get functional trait data for all the species that are in inventories, like the U.S. Forest Service inventory analysis, where there's hundreds of thousands of plots all over the continent. And if you have the functional trait data from herbarium specimens for all of those species, and you develop ordinations and get axes of variation based on those plots and their functional traits, and then you couple that with satellite data that overlap with those plots, and they're geolocated, the satellite data, the pixels from the satellite data are geolocated with the plots. You can then use the satellite data to predict the ordinations using the functional trait data. You can also use that, do the same thing with phylogenetic information, which means you can then map the functional trait information and get map phylogenetic lineages. And the satellite data is continually being generated. So you can actually get change through time. You can get functional variation, functional composition, phylogenetic composition through time doing this, which At a time when we are losing so much of the world's biodiversity, this gives us a way to work towards the global biodiversity framework targets, which are getting us to think about conserving biodiversity. How do we verify that? Well, we really need satellite data that we can interpret in terms of biodiversity. Also, restoration of degraded ecosystems and payment for ecosystem services. These kinds of things require verification. So satellite data is the way we can do that. If the biologists work really hard with those folks to make sure we can interpret that well. So a lot is being invested into these satellites, both around the world, NASA and the European Space Agency and other space agencies, and let's just hope that continues. In addition to scaling up, we can also move across evolutionary scales. So this is coming from a NIMBIOS working group from a while ago, and Dudu led the work coming out of there. But we combined fresh spectra from hundreds and hundreds of plants around the world and looked at the phylogenetic signal in those spectra. And when we look across the whole tree of life for plants, there's phylogenetic signal in all of those wavelengths. We can then move into particular regions of the phylogeny. We still see high phylogenetic signal. We can even go down to the population level, and now we see, there's only some regions where we see this kind of signal. But we can look at how spectra vary among individuals within populations, among populations within species, among species within clades of larger and larger size. So it's also a way of scaling through evolution. And this is using radiative transfer models, a simulation for number of layers or leaf structure, using 2 different evolutionary models, simulating the evolution of that structure through time and how the dried leaf spectra change through evolution, and what their tip spectra look like for these 2 different models. And this is a paper that I've been working on for, I don't even want to talk about how long, but this is modeling the evolution of plant function from spectra for the oaks and looking at how those plant traits relate to the different environmental niches in which oaks occur and how their trait optima shift over the course of evolution of this clade. And here's a study that postdoc Mariana Hernandez-Leal, who's currently in Mexico, and I'm hoping she comes back. She just submitted this paper. This is a group of oaks, a monophyletic group of oaks, 7 species that occur throughout the southeastern U.S. and Mesoamerica. And she finds that spectra work really well to discriminate among taxa. So the diagonal, if it's a dark color, means that there's very high accuracy in classification and differentiation of species. So this is using partial leaf squares discriminant analysis with pressed leaves. And she's also using that spectral information to compare to genetic information to test for selection using QST, FST analysis, or in this case, a PST, FST analysis. So these are the kinds of things that can be done with spectral information. She just used the leaves that I'd saved from all the genetic work and measured spectra on all of these. And so it's a new use of material we already have. And then we can do taxon classification at higher and higher phylogenetic scales. So this is again from that NIMBIOS working group that this is, and reanalyzed from all the work Dudu did on this. This is using fresh leaf spectra and at these very broad clades. The diagonal again means very high accuracy in classification. So we can use the spectra to put specimens into different broad clades. And you can't do 90,000 species all at once in a single analysis, but you could do this many species and get high accuracy. And then this is looking at orders. So once you've got them grouped into broad clades, then you can look at orders And then within orders, you can go down to families. Some of them work, and some of them don't. And Barbara's going to talk more about this. And then from families, you can go down to genera and species, like I just showed you. So this is using a stepwise approach is one way that we can enhance we can use this information for classification if, in the case where there are specimens with unknown identities and there's perhaps not reproductive information, so there's no other way to try to hone in on where those unidentified specimens are. Okay, but here's what we're going to spend a lot of the rest of today on, are all of the myriad challenges. So to do any of this collaboratively and to be able to aggregate what we're doing across institutions and over time, we need to agree on methods and metadata, instrumentation, protocols, metadata standards. We're going to have to sort through all the complications that arise from specimens, which are different from pressed leaves or fresh leaves, and have variation in leaf size, developmental stage, collection environments, how they're treated when they're stored and moved, the storage conditions, the background that's used, the glues. And so all of this is going to require collaboration, and we're going to have to figure out how to do this ethically, make sure people get appropriate credit for the work they do and things are done fairly. We're going to be hearing about all of this. So here we saw this yesterday, just the glues that go onto specimens, the glues contaminate the spectra, and so this is just something that is the case for most specimens. Would be really nice to have a black background because the paper and anything under the leaf will contaminate the spectra. The light goes through the leaf and is reflected back, and if there's something like paper, fiber, it's going to change the spectrum. So having a black background is ideal, but many specimens are really stuck to the black background and to the paper, and there's no extra tissue available. So what do we do? Some leaves are really difficult to measure, and these aren't even the worst case scenarios. So we're going to have all of those kinds of issues. Do we measure adaxial and axial sides of the leaves? Spectroscopy has been used for remote sensing for so long. The main protocol is just to measure the top surface. But for herbarium specimens, I mean, we're going to want to do things differently, so we're going to have to depart from previous protocols. Developmental stage is going to be really different. These are oaks that are flowering, so we know the leaves are not fully expanded. And then how do we keep track of which leaf we measured? So we'll talk about this is the method that you use, Barbara, right? So we'll talk about this. We're all using different equipment. I have 2 minutes left, but I'm going to be okay. We're all using different equipment. As Dudu can tell you, it's kind of a nightmare to bring data together from all these different instruments. It can be done, but it's not trivial. So we're going to have to think about how we do that. The setups are different. Maybe we can be really clear on the optical setup so that we harmonize as much as we can from the outset. Okay, so here's what I hope we get from this meeting. I hope that, well, we're coming together as a global community of folks who are enthusiastic about herbaria to do good work together and to help each other. And I really hope we can agree upon minimum standards for protocols and metadata, that we walk away with that, something we agree on as a minimum set. That we learn from each other and stimulate collaboration. There's so many opportunities in this group. And have fun in these tricky times. Let's just have fun being here together. And then I'll end with this. So this morning we're having these thematic talks and then lightning talks that will be followed by a panel discussion. We're going to do a quick photo out in front if the weather holds up. Otherwise we'll do it in the building. Lunch. Then we're going to go back here for this protocol and metadata discussion. We've got instruments set up and people have worked hard to prepare this discussion. Dinner tonight, 5:30 at Barcelona's. should be fun. Tomorrow we meet at 9 a.m. The bus will come to your hotel, and we'll have a tour at the Arnold Arboretum. There'll be a brainstorming session out there, and 3 p.m. the bus returns back. So that's what we're doing, and I just want to make sure to thank Jeanette Everett, who's not here today, who did so much of the work helping everybody get here, Dawson White for taking care of so many of the logistics and running the virtual part of this program, Antonio, Michaela, Jonathan, Nicole, thank you so much for making this happen. And thanks to all of you for coming all this way to be here. Just really grateful. Thank you. You get to introduce yourself?
Yes, I can.
Extending this effort to countries that don't have the resources to...
Oh, the pointer. This button. Okay. And then press
Yeah, because the equipment is expensive. To be discussed. There'll be ways to deal with that, I think.
First, thank you for the invitation. It's really glad to be here with you and present our results with our studies with the spectra. My name is Flavia Durgante, and I'm from Brazil, south of Brazil. And I'm work at INPA, National Institute of Amazonian Research, for more than 15 years. And now I'm living in Germany, work in the Karlsruhe Institute of Technology. And today I will present some results that we have with the spectral herbarium from INPA, equipment methods, and application to species identification. Okay, as we know, in the Amazon forest we have high diversity. It's hard to work with that in the field, and the authors are discussing about how many trees we have there. And some authors are saying that we have around 6,000, others say we have 11,000. The estimative is 16,000 species, but the truth is a lot, and we need to work with this high diversity, and we need some techniques and conditions to work with this. And in that case, since 2009, we have been using visible and near-infrared spectroscopy to create a spectral herbarium that can assist the botanist to recognize species. And at the same time, now we are moving to field that we can recognize living trees in field that can help us to improve the forest inventory as well. We are a group of researchers, like I can to highlight Alberto Vicentini, Flavia Costa, Mike Hopkins, Niro, Isolde, many researchers. We are working there. Since then, we have many master and PhD students doing some tests with this. And I think until now, we have more than 30,000 spectral reading specimens that we are studying, okay? During this period, we used different equipments. The first was for FET and IR. Is this equipment here that is measuring for this wavelength 1,000 to 2.5 thousand nanometers. Is more used in the chemometrics group with pharmacy and chemical analysis, but we use it in field, in our samples. You can see here the leaf, and we have the black material in the background that we measure. After that, we used and tested the AICD field spec. I know the picture here is in field, but we use this in herbarium samples too. And this equipment is measuring the visible and near infrared that we have 450 nanometers to 2.5 nanometers. And now we have a new project that we call Spectra Pop that we are trying to popularize, to use a small micro NIR that's cheaper comparing to the others equipment and can be more popular, that people can use more. And we are trying to test how good it is to move from good equipment for one that's not so good, because we are losing resolution, we are losing the range of the spectra as well for these equipments, a short area that's measuring. Okay, the first test that we did was with the Lecitidaceae family, that we have the hyperdominant species in the Amazon, Eschelera coriaceae is from this group, from this family. And our first test was to see if we can separate Eschelera coriaceae from other Eschelera closely related species. And to do this with high quality and accuracy, we did this first test. Can the spectrometer see the group of species that Scott Morris saw within this genus? Because Scott Morris was the specialist of the family, and he went to Manaus and worked hard there with 100 hectares permanent plot. And I wrote to him this time, and he told me, include Corythophora genus because it's a monophyletic group, and Eschelera is non-monophyletic group until now. And he told me this is not working, but you can try, and we did. And in that case, I used his samples that he collected. I decided, okay, I wanted to know what he's looking, because if we include more people, we include more errors. And we used samples collecting since 70s until 2010. And we used this equipment that I told you from this region, 1,000 to 2.5 thousand nanometers. And then we tested how many readings I needed to take this signal from the specimen. And we measure 36 readings in total, 6 leaves, 3 readings in the adaxial side and 3 in the abaxial side, because I don't know what's the best way to measure. And with this, we use the linear discriminant analysis that I have the absorbance values here in the dependent variable to estimate the name of the species. This was the model. And I tried many models, only abaxial, only adaxial, 36 readings, or only one spectra, or we try to answer how many readings I need to take this signal from the specimen signal. And our first result was really good, and we published this. And we recognized the groups that Scott Morris saw to Eschelera with 99%. And with only one spectra, we have 97%. And then we try to test how many readings is necessary. And we used the mean of 50 replications, dividing the 2 sets of data, like we have some data to calibrate the model and other set to validate the model, and testing many readings from 1 to 36. And we saw that 5 readings is enough, don't have significant difference to 36. 5 is good. 1, it's possible, but sometimes we lost a lot of information only because we have low number of readings. And then we beginning to dream, oh, maybe we can try to build a spectral herbarium. And for us, maybe if we can include or create our Duke Reserve book, this is our Bible, to put inside of the computer maybe can be helpful for the people who is working with the high diversity. And then Alberto Vicentini advised Carol Curti in master thesis that they adding more species now, because first I tested only with one genus, and now we tested 34 families, 52 genera, and more than 100 species, and they recognize so well this group of species. And then Flavia Costa advised Carla Lange, and now we are trying to understand the ontogenetic signal. For example, if I have a model of only adult trees, it's possible to recognize seedlings, because sometimes the morphology change in the ontogenetic time, and she did this test, and she saw that probably we need adults and juvenile trees inside of the same model. The adult models is not so good to predict juvenile. Young trees can be better, but maybe it's good to have this variation inside. And then we're beginning to try to understand the geographic signal, because if I want to build a model or to use the IMPAS herbarium to create this big model, how useful is to recognize species from other side. And here I had the opportunity to go to Paul Fine Lab in Berkeley, and we collect many samples there, the spectra, and we tested this question. It's possible to use a model from samples only from Peru to estimate French Guiana samples. And here we have only 4 species, and is a protein group. Protein is not so easy group to test methods. This is good to say, but we saw that we need more variation inside of the model. And then Mike Hopkins and Niro and me, we advise Isaelene Saarto that continue with protein. And now we are trying to test if I have samples only from Manaus, collected around Manaus, I can recognize the species in the other regions in the Amazon. And I can show you here. Here is Manaus model trying to recognize the species from other sites. And in 2017, she recognized only 66% of the samples. But in 2020, Douglas Daly revisited the genus, the group, and he changed some genus and names, and she improved the model. As you can see, we need very good groups to test the spectral data. And then Paulo tested some clones from Bertolacci Celsa too and distinguished so well. Alberto Vicentini and Eduardo Prata, they confirmed that the spectral data and DNA data can support species hypothesis. And then Caroline and other researchers are using the spectra to corroborate with species hypothesis, the new species hypothesis there, because they have only morphology, and now they have morphology and spectra. And as I told you, we are testing this micronear now, and we are finding good results. As Jeannine explained, here in the middle is when we have a good signal. And here is the expensive equipment comparing to this equipment. As you saw, we are moving from same genus, species, and DNA to help us to delimitate new species. Now we are here organizing herbarium protocols. We think that the spectra can help us to improve the communication between herbarium, because we can to change now chemical data to not only pictures, and we can to to use the spectra to help to clean some errors. And the idea is to do this many tests to improve the quality of biodiversity inventories. And thank you so much. I'm open to collaborate, and if someone wants to continue this talk, I'm open. Thank you.
Thank you. This is a really interesting talk. I was wondering how you treated the abaxial versus adaxial data. Did you just put it all in the same model, separate models?
Okay, we did separate it first to understand how is the best size to recognize the species. But now we are using both sides together, like making the average. Average of 36 readings is the specimen signal. Because I saw that during this period we did it many times, and some groups, the adaxial side is better than abaxial side, or opposite. And sometimes we only can read adaxial side. Because this we are trying to measure both sides, that, for example, if you have only adaxial side, you can take only adaxial data and create your model. And sometimes the adaxial side has more information that can help us to distinguish species.
Okay, thank you.
Do you think your work will determine the total number of tree species? Earlier you mentioned about 11,000 versus 6,000. It's a big difference.
Yes, I know. 16,000 is the estimative that how many species are. Probably is more. I think our work can help us to recognize the diversity faster, because sometimes we have only morphological signals that can be different species, and now we can use this chemical information to see, oh, it's totally different groups, and this can be faster and help us to get more funding to do DNA analysis that's not so easy for us in Brazil. Yes.
And your images showed members of the collaborator community measuring tree bark, as well as leaves.
The quality of the tree bark. Yes.
The bark. So were you actually measuring multiple plant parts?
Yes, we are measuring bark, especially in field. We have very good results to recognize species by bark spectra. And our idea now is trying to create some quality assurance, quality control to permanent plots. Like I can go there and measure all barks and say, your inventory has 80% of matching the names that you are saying. And this can be together with, oh, if you have less than 50%, please go there, collect this, and go to the herbarium again and improve your names. I think this can be the future, and I'm looking for this. Finish? Thank you.
Hello, everyone. I'm Meireles. I'm at the University of Maine. I mostly do evolutionary biology, as you can kind of tell, but I'm not going to really talk about any of that today. What I'm going to try to do is really dig into herborization, its effects on spectra, and then what we can do with those data. So it's fair to say that we're all here because we're interested in plant diversity, right? We want to understand it. We understand, plant identity. Maybe you want to understand why plants live where they live or how they function. And I really like this image because it highlights how dynamic, diversity really is. But what we typically do is that we go out in the field and we take these snapshots of diversity in the form of a herbarium sample or a specimen. We do all sorts of things, right? We lab glue on them. We have them sometimes on non-acid-free paper. And you let these guys age for a very long time in a herbarium. So I think it's important to recognize that these snapshots are fading snapshots of the diversity we saw previously. In addition, when we are in a herbarium taking spectral measurements, as you can see Cornelius and Kira doing here, you may be faced with a challenge that Jeannine was talking about, right? Maybe you have specimens that are glued down, and you can't do anything about it. You have to measure them that way if you want that plant. The paper's going to show through. There are lots of contaminants to that spectrum. So I think there's a giant distance between what we do with spectroscopy in a herbarium and the reality of the world, right? And a fundamental challenge, in my view, is to understand how we can close that gap, how we can make that, a little more of a direct comparison. So that's kind of what I want to try to talk about today. Here's the outline. There are two major topics. The first one is assessing how aging and then these contaminants, paper and glue, affect the spectra and the kinds of inferences we can make, based on the spectra. I'll just do classification. And the other important topic is trait estimation. But again, I think I'm interested in estimating what traits were in the field when the plants were fresh. And the second thing that I want to talk about, and is also linked to that first, beautiful fall image, is to acknowledge that plants are dynamic. They change through the season. Their attributes change through the season. So how can we, take that into account or possibly leverage that information to learn more about plant diversity, the dynamic aspects of plant diversity. So let's jump into the first one. And this is all embedded in an experiment, a herborization experiment that we've been running at the University of Maine, where I'm the director of the herbarium, since 2020. We have 27 different species. We have ferns and conifers and flowering plants. And we've been monitoring or taking spectra, as a time series since 2020. In addition, we actually have mounted those specimens, right? So we have leaves in packets. We have leaves that are loose but are actually in contact with the herbarium paper, and some of them are glued. So we have all that variability that we're trying to keep track of and assess, their effects. So the first question, or the first thing we try to tackle, is to understand how herborization and aging affect these spectra. And I'm not going to dig super deeply into that. I'm just going to show a few examples. So here are 6 different species. There's interrupted fern, there's a hemlock, cattail, all sorts of different things. And in red, you see the loose leaf spectra, and the other 2 are spectra that are, either against the paper or glued down. And the basic idea here is that, yeah, there is a very significant difference. There's a very clear signal of the contamination that Jeannine was talking about. It's interesting to see that because of the physics, the degree of contamination varies with how thick the leaf is. A really thick leaf will not show a lot of background. A really thin leaf, think something in a Hymenophyllaceae, in a, one cell, you're mostly going to see paper. The other side of this is aging and that process of aging, right? So here we again have 2 of the species. In red, and I'll step out of the mic for a sec, sorry. We have the fresh spectra, right? So this is when things were collected. In blue, this represents a spectra as things were drying, so within a week of collecting or and drying up. And then the other measurements are all measurements of this time series that I've been talking about. And these are the loose leaves, by the way. So you can see, again, very significant effects of aging. But I think the most critical thing to realize is this is different for different species, right? So the parts of the spectrum that change are or change more are different in different taxa. So in white oak, there's a lot of changes in the visible. In red maple, lots in the infrared. So the y-axis here is a coefficient of variation, just the amount of variability normalized by the mean. Oh, I think these were missing a few slides. It's okay. So I'm actually going to jump into, okay, we saw that these spectra are aging, that there are contaminants. How does that really affect the kinds of inferences that we want to make? So here's an example of a classification model that we built. On the left-hand side, you see loose leaves in the 4th year of being herborized. And it's pretty good accuracy, 97% accuracy. You see that diagonal is mostly what you have. But if we actually take loose leaves and glue down leaves and model them all together, and instead of not measuring those glued down leaves, you have an indicator, right? A covariate saying those are glued and those are loose. You can still do pretty well, right? You can still do classification with these glued down leaves. Now we're jumping into how do we fare when we try to do trait estimation. And here's so these are going to be a little hard to do from here, so I'll jump over there again, and hopefully you'll pick it up. I can't do that. So these are, on the x-axis, we have the true values of these, of a bunch of different traits: carbon, nitrogen, LMA, and chlorophyll. And on the y-axis, we have predictions. And these predictions are based on, the pressed leaf model that Sean devised. And we can see that the models are not doing all that well. They're pretty far from the one-to-one line in this specific case, right? And that's true for both loose leaves and for glued leaves. It's worse with glued leaves. So, you know, what do we do? Do we throw our hands up in the air? Well, because we have that time series, we can actually develop a new model. We have the true traits. We can model the traits directly with the spectral data from the herbarium and the fresh traits that we measured way back when, and develop models that actually do pretty okay. I mean, I'm not, you know, adding any of the stats here, but you can see that they're all on the 1-to-1 line roughly. So we can do pretty well. You know, there is hope to actually assess the traits of when these plants were fresh by measuring herbarium spectra. If we build a library, if we have a herborization experiment that is thorough enough, and that's my argument, I think. And again, 2 minutes? Okay. And again, if we do, if we mix up glued and pressed leaves, we can still do pretty okay. So there's a lot of hope, I think. And this is a last bit of the talk. This is work that Cornelius there has done. He measured more than 30,000 spectra for this. So, yeah, he started his PhD like 30 years ago. It's been a long way. But this is the idea of like traits are dynamic. Like things are changing through time. There's trait variability over the season. I mean, Chuck is all into that, right? And traits correlate differently. So the idea is, how does that affect our inferences about function when we go to the herbarium? You know, we don't necessarily have the luxury of saying, I'm only measuring things that are peak season. For some plants, that's mostly of the material is going to be senescing or early. So let me just run this through real quick. 3 different species. One trait, in this case, LMA. We have several others. And you see in blue, you have the true traits measured in the field through the growing season. And in orange, you have predictions from an already established PLS model. And basically what we see is, as senescence starts to happen, both here and over here, the predictions get a little worse from an established model, because those models were fit, with peak season attributes. Similar story for chlorophyll. I mean, there's a bunch of, the models are predicting negative values of chlorophyll towards the end of the season, right? That's physically impossible. So it means that, we have to do something. We have to work harder to build models that actually encompass all of that phenological variability so we can do better. I have, I've had here like an explanation for why the models may be misbehaving, but the bottom line is, there's a lot to do.
This is where I get to
okay.
off the stage too.
Sorry.
This is what happens.
That was it. Yeah.
Everybody, thank you for coming. I want to thank Jeannine and the whole staff of Harvard for making this possible. To be honest, I wasn't sure it was going to be able to happen, so I'm really pleased to be here with also inviting, being so inclusive is such an important part of this process. So thank you very much. I'm going to shift a bit of gears and talk to you about tropical rainforests. I'm really tackling the near-infrared scanning approach from this perspective about how we can understand the evolution of tropical rainforests. Just as a quick background, tropical rainforests are one of the most biodiverse terrestrial ecosystems on the world. Some estimates suggest that we have over 50% of all known plant species occurring in these rainforests, which only occur on 70% of the land surface. They also provide besides being a biodiversity reservoir, they also provide extremely important ecosystem services, in particular such as climate regulators in terms of precipitation, and not just at local levels, but also at global levels. So the preservation of this ecosystem is extremely important at multiple scales. But one of the main questions that evolutionary biologists have been asking over the years is how did this huge diversity come to be? What are the drivers of this rainforest? And these questions have been ongoing ever since Alfred Wallace wandered around the Amazon and the Malay Peninsula. So there are multiple number of hypotheses that can explain this. I'm not going to go over all of them. For example, some of them have to do with the tempo of diversification. Alfred Wallace was one of the first to suggest the hypothesis that actually rainforests seem to be extremely stable in ancient ecosystems, and so probably there were low extinction rates. He didn't really use these terms, but that's kind of what he was alluding to, and that this led to a constant accumulation of diversity through time. And if you link that to the old age of rainforest, that would be one of the explanations. But we all know that tropical rainforests are extremely complex and dynamic ecosystems. They've been varying in terms of size and fluctuating through time. And this led to some other hypotheses being suggested, for example, that there are actually lots of opportunities to have bursts of speciation linked to interactions, for example. And that led to hypotheses suggesting that there might be ancient bursts of speciation explaining this diversity, or more recent ones. And these hypotheses have been tested using molecular phylogenetics. And here you have an example of the genus Guatteria, which is a large tree genus that was suggested to be very recent and one of the examples of these bursts of speciation. Another set of hypotheses, or there are a whole bunch of other ones I'm not going to go over, but is a very important one that was suggested by Elrich and Raven in the 60s, was this biotic interactions, and in particular was herbivory. So herbivores and plants co-occur, obviously, and in tropical rainforests, this interaction is extremely important. And so this kind of led to this idea that we have this arms race going on. A plant is going to try to evade or escape the pressure of a herbivore, probably by trying to evolve, in one aspect is trying to evolve chemical or secondary compounds, which is going to make the leaf a bit indigest for the insect. And when it does that, it kind of enters an adaptive zone where it can radiate. And that kind of led to the ideas of escape and radiate hypothesis. And so this is an important hypothesis that is generally being tested at local levels, in particular using genera such as Inga as a very important role model. In the project that I'm leading, and we're in the 5th year now, we were interested in trying to address the hypothesis of rainforest evolution, but really from a global scale. So initially, a lot of people were looking at this at local scales within a continent, and we were really interested in trying to scale this up at the global scale. And for this, we decided to work on the Annonaceae family, which is obviously a family I know well, but Annonaceae is also a very important tropical plant family. It has about 2,500 species, which is diverse, and yet it's still a family we can kind of tackle within a 5-year frame of a project. What's interesting about Annonaceae is that it's very diverse chemically. So Annonaceae belongs to the Magnoliales, and one of the field characters of identifying Annonaceae is crushing a leaf and smelling it. And so this kind of translates this idea that they have high chemical diversity, and a lot of chemical analysis have shown a lot of alkaloids, a lot of phenols occur in Annonaceae. So this could translate an idea of some kind of arms race ongoing in Annonaceae. It's an ancient family, it's Magnoliales, so the family originated about 90 million years ago, which kind of coincides with the origin of modern-day tropical rainforests, although that claim is a bit debated. But what really makes Annonaceae important is that we have very solid taxonomic background. We're really standing on the shoulders of giants here for many decades. A lot of my predecessors and ongoing colleagues, some of them here, we've been working a lot on understanding the taxonomy of Annonaceae, and it's probably one of the best known tropical plant families in terms of its taxonomy. And so within this project, we wanted to generate novel data, so sequencing a lot of specimens, but also to generate complete data, which meant that any kind of data we wanted to generate, we wanted that to be applicable to all species, or as much as we can. And so for that, we turned to the Herbarium herbarium specimen. The herbarium specimen here is really the center, the star of the project. From Herbaria, we can extract a number of things that we all know. Morphology, we can extract now DNA. Thanks to next generation sequencing, we can extract distribution information, which is useful for ecology. But what concerns us here is that we can also now use near-infrared scanning to try and extract some kind of proxy of chemical diversity and structural diversity. So I just want to say that this project was a very long project, and we spent numerous times sampling Herbaria. I just want to shout out to all the collection managers who allowed us to destructively sample their specimens for DNA extraction. You have an extract of that. This was mainly led by Carlos Rodriguez, who spent a significant amount of time sampling in Herbaria. Then the samples were sent to my lab in Montpellier, and my colleague Vincent Soulet did all the molecular biology, and this was mainly done during the COVID, and he was one of the only ones who was allowed to enter the lab during that time, so it was a very lonely and long process for him. So I just wanted to shout out to these two for the incredible job that they did. Anyway, long story short, we generated one of the first species-level phylogenies of a major tropical plant family. As you can see here, we have about 83% of species sampled, and we're doing all kinds of tests of diversification rates and understanding how rainforests evolve globally to locally across scales. But what we wanted to do, and so when I wrote up this project, I was, okay, how can I somehow test the herbivory hypothesis using herbarium specimens? And so I naturally turned to near-infrared scanning. So I'm really a newcomer to this, and I really also stand on what a lot of people in this room have been publishing over the years, and I just wanted to thank them for all those inspiring publications. So what we did is we just set out kind of really bluntly and said, okay, any specimen we sequence, we're going to scan using near-infrared. We acquired an ISD lab spec. And then Carlos, again, as he sampled, he also scanned the specimens. And so today we have about 1,740 species scanned, representing about 70% of species and 96% of genera all across the world. As you've already seen, here's an example of a spectra. We would scan the adaxial side of the leaves, and we would sample 20 times every specimen. So here you have the 20 in light gray, the 20 spectra, and then we would use the mean of that and normalize and do all kinds of other specimens. So this part here is really the work of Khalil, who's here today, and again, thank you for inviting us. And so we're really at the beginning. It's been some time, but it's so much data, and it's also new for us that we're trying to learn as we go and interacting with this incredible community. If we have some very basic looking, so if we look at all the spectra we gathered for Ananaceae and we look at the standard deviation, we can see that some areas are more diverse than others, in particular here in the shortwave infrared parts. This kind of variability stands if we look at the subfamilies, and then it also stands if we look at the tribes. So we also have these different regions that are highly variable. Some tribes like this one, which is a very small tribe, actually seems to have huge variation in that level. Before Khalil came, actually, one of the first students who worked with me on this project, so as a newbie, we both sat down, and this is Linny Bennett. She was from the US. She did a master's in France. And we said, okay, she was really interested in evolutionary rates. So inspired by work from Dudu and colleagues, we said, okay, we have a phylogeny. We have spectra for 81 genera at the time of the project. And she looked at phylogenetic signal. We saw that we had similar results to phylogenetic signals that were found at angiosperm levels. And then she used this and used specific macroevolutionary models to try and plot this onto the genus level phylogeny. And what you can see here, Ananaceae has two large subfamilies. One subfamily here, Marmoides, which is characterized by these very, very short branches. It creates a huge amount of problem in terms of taxonomic resolution and a rapid, what we could assume some kind of rapid evolution into Southeast Asia. And then we have this subtribe here, this subfamily, which is much more diverse in terms of morphology, much longer branch lengths. And when she looked at the 27 most phylogenetic significant wavelength, we found that this area actually had lower rates of evolution, and it was pretty homogeneous, which kind of fits with the idea of what we get from taxonomy from this family. And in contrast, the larger Ananoides down here has highly heterogeneous rates, and most of the time these rates are higher. So again, kind of going towards what we know about these evolutionary rates within Ananaceae. With Khalil, we're trying to analyze all this data now that we have a complete phylogeny and go a bit more down with more appropriate data sets. I'm just going to finish in terms of spatial analysis. So what we can do, and my PhD student Serafin Streef was working on spatial patterns within Ananaceae using the phylogeny, and he generated a species distribution model map of a species diversity of Ananaceae based on a huge database, which was obviously taken from Herbaria. So Khalil used this data set and used the NIRS information to generate a spectral community diversity map. And this is all very preliminary, it's all relatively straightforward, but it has a very marking pattern, which is that we have higher spectral diversity and distance in Africa in general. And this really goes a long way with what we already know. There was a recent paper in Nature which also suggested from remote sensing, which also suggested higher spectral diversity in Africa. But if we come back to Ananaceae, one of my postdocs, Wei Tsu from China, he was working on a functional trait database. So he was databasing functional traits, lianas, leaf sizes, all that. And he synthesized all this. So this is the raw map of species diversity from our herbarium data set. And what he found is that indeed Africa is also showing higher functional diversity. So this goes back to what Jeannine was saying at the beginning, that these spectra really seem to be translating something also at these evolutionary scales, but also at local scales. And so again, with Khalil, we'll be trying to investigate a bit more what we can do with this type of data at this scale. I just want to thank all my team. Anything that I say here is all thanks to them, this incredible team of people, hardworking. The Ananaceae Global Phylogenetic Consortium, it's a large consortium across the world where we've been collaborating. And also want to thank Sirad, Samantha Bazan, who's here, for sharing initial ideas of protocols of scanning, which really allowed us to hit the ground running, thanks to their expertise. And thank you for listening. Huh. I'm not going to answer that question. It's a complex question, and we're having some surprises with palms, and we're having, but yeah, it's a long debate. I'm really happy to talk about that because I know that we exchanged about that at one point. But for me, they kind of originated about 100 million years ago. But then you have proto-rainforests and actual rainforests, and fossil record seems to suggest that at the Cretaceous there was a big change in how these rainforests looked like. And so the modern structure probably originated at the beginning of the Cenozoic, but at least the assembly of these taxa together and forming some kind of rainforest type environment was probably earlier than that. It's a long debate. Thank you.
Well, good morning, everyone. I'm Pam Soltis. I'm a curator and professor at the Florida Museum of Natural History at the University of Florida. And this has already just been a fascinating workshop so far this morning, and I'm already learning so much. So many thanks to Jeannine and her team for organizing this, and also for inviting me to participate. So herbarium specimens are fascinating for so many reasons, and one of the things that I'm really drawn to collections for is because our natural history collections provide us with opportunities to investigate unanticipated uses of these specimens. So many of our specimens were collected decades or centuries ago, and yet now we're seeing how we can be putting these specimens to many different uses, including the uses that you're all working on in terms of spectra. I don't work on spectra, but I'm fascinated by the topic, and I particularly am excited about the sort of multiple scaling levels that Jeannine mentioned in her introduction. But instead, I am the director of research for IDIG-Bio, which is the National Center for Digitization of Biodiversity Collections here in the U.S. And in this role, I'm actually not funded to do research, but I'm funded to help facilitate research using the collections that have been assembled, and particularly the digital representations of these specimens. And so I'm really excited to think about how we can develop new methods of integrating data and of pulling together new ways of digitization and imaging so that we can generate new types of data that can help us address a whole range of questions, particularly those of societal relevance. Now, I realize that many of you, not being from the US, are probably not very familiar with IHerbBio. And so in order to discuss and think about how we might be able to database these spectral data once we get to that point, I thought it might be valuable to provide just a little bit of background on IHerbBio. So you can get to IHerbBio by going to iHerbBio.org, and this is actually a database. There's a portal where you can access the information for all of the specimens across taxa that have been digitized and submitted to IHerbBio. So from that website, you can see a heat map. We have not only U.S. collections, but also some partnerships with other repositories in other parts of the world. And so we have shared data, for example, with the Atlas of Living Australia. So we have pretty good representation of Australian collections as well as U.S. collections. And if you're interested in actually seeing what we have, there's a search portal. You can search by any sort of taxonomic measure. You can search by geography. You can search by collector. You can search by dates, et cetera. And so you can set up your own search parameters either right there in the portal, or you can use the API and several R packages to actually go in and specify what you're looking for and get the data. So for example, if we wanted to ask about red maple, we could just type this in as a scientific name, and then we would be returned a set of records. You can do this either through a download of data or right there in the portal. And you can get, for example, a map that shows where those digital, where the georeference specimens are. And you can click on each of the dots and you'll be, you can pull up a thumbnail of the record and then go to the complete record. And in many cases, those complete records will have images associated with them. So when we started IHerbBio, and I'll say right now we're in our 14th year of a 10-year program. We actually have one more year of funding, but then NSF has said that's the end. This was supposed to be a 10-year program, and we've funded you for 5 extra years. So we're working on sustainability measures, but that's maybe another story. But when we started on this, GBIF only had 16 fields. It didn't take images, and so eventually GBIF then restructured to be able to accommodate the sorts of information that were being generated here in the U.S. through this digitization project. It was a $100 million 10-year project to jumpstart the digitization of natural history collections. But IHerbBio is just one data aggregator. There are many others around the world. Several of these are regional. Some are national, some are taxon specific, for example, VertNet. And all of these, or just about all of them, feed into GBIF. And so GBIF represents this global biodiversity information facility. And it assembles information from all of these other aggregators. And so to get a snapshot of the sorts of collections and observations that GBIF holds, this is just a view from last summer. But I will make the point here that many, in fact, most of the occurrence records that are now reported in GBIF are actually observations and not specimen-based. And so, of course, you can parse by basis of record and only pull up the specimen-based ones if you're interested. Now, if you are interested in seeing what sorts of information might be present in any of these databases, I took my search for red maple for IHerbBio and downloaded the data. We actually have 82 fields. Many of these are Darwin Core fields. That means they follow the Darwin Core standards that have been established and approved by the Biodiversity Information Standards Group. But some of them are actually GBIF-specific fields, and some of them are IHerbBio-specific fields. And so there's a whole range of these fields, and in order for databases to talk to each other, they have to share that structure. And the most common of these structures is the Darwin Core set. And so I've just indicated a few of these here. So there's Darwin Core basis of record here, the catalog number, and the collection code. And this is just, as I mentioned, just a few of the 82 fields that would be outside, well outside the building if we had extended this table. So in order for these different databases to be able to talk to each other, or for people to be able to download data and integrate the data, they need to, the data need to share standards. And the Darwin Core standards seem like the best way to go about bringing about the ability to share spectral data. And more on that in just a minute, and also later today. But one of the things that's really clear when we start looking at these specimen records is that the specimen records primarily are a snapshot of what's on, in the case of plants, the herbarium specimen label, and maybe a little bit of additional information. We know, though, that there's a lot of other information about that specimen that's captured in perhaps other databases. And so I really like this cover image from the book edited by Mike Webster on the extended specimen, where it clearly shows the specimen here in the center, but some of its derivatives. So, you know, maybe there's a parasite, there are pathogens, there are DNA sequences, isotopes, and even, for example, species distribution models that were generated, including data from this specimen. And of course, none of that information is actually in the specimen record itself. That information is often, well, maybe somebody's freezer, maybe on somebody's hard drive, maybe in another database like GenBank, but it's very difficult to actually pull all of that information back together. So this digital extended specimen is getting a lot of attention in terms of concept, but in terms of real progress toward being able to pull together all of those derivatives back to the specimen, we still have some challenges. But a working group here in the US has been working in parallel with similar groups elsewhere globally about how to extend those collections. And so this is just a report on extending the US biodiversity collections. But as I say, there are now global efforts to think about how we can pull these data records and extended specimens together on a global scale. And this just shows an example from this report about how we can start out with the physical specimen. Well, first the collecting event, then the physical specimen, the digital specimen, and an image, and then all of the things that can be derived from that basic original collection. So how best do we represent spectral data obtained from herbarium specimens? So we've already heard about the different emphases that people have in their work on using spectra, sometimes for species identification, sometimes for studying herbivory, sometimes as another representation of functional traits, and so on. And so there may be multiple different aspects of the spectral data that may be of interest across this very broad global community. So how do we actually capture all of that information into a set of Darwin Core standards and extensions? And one thing that I'll note, and I definitely don't have the answer to that, that's why there's a subgroup working on this, and I'll be really interested for, and I'm looking forward to that further discussion on some of this. But I will note that once a Darwin Core extension is developed and approved, what could go in that field could actually be a URL, and that URL can then go to another database. So for example, in that field that, or snapshot of the fields that I showed before, one of them was the collection code. For many of those collection codes, it's a URL, and you can go from the URL, then just click on that in the field, and it'll take you to whatever the representation is that that particular collection has provided. And then you can drill down into the information and the data more deeply. And so I'm just throwing this out as a potential way of making connections between what goes into a field in a database and what then is used to represent all of the data. So let's say there is such a way of doing that. We have Darwin Core extensions. We have a way to capture this information digitally in a way that databases can interact with each other. How do we actually then link the herbarium records with the spectral data? So of course, if you're starting out with new collections and you maybe capture your spectra prior to mounting the specimen and then recording the data, all of that information could go into your local herbarium at, in one fell swoop. But in fact, a lot of you, as the discussions have been going, are actually capturing data from herbarium specimens that have already, probably for the most part, been digitized and that have already been submitted to either a local aggregator or to GBIF. So how do we actually add those spectral data to those herbarium records? So one possibility is to just put those spectral records and data into the notes field that a database already has. So this is sort of an option that some people have used for other sorts of information, like what are the specimen character, or what are the associated species that are present in with the specimen. A lot of observations, ecological information, and so on may be placed into the notes region. That gets the information into the database and could go from both, say, the Harvard Herbarium database to IHerbSpec and GBIF, but of course it's not searchable if it's not easily searchable if it's found in the notes. But it would be a potentially easy way to do this in terms of just getting the data out. So the data could be in the notes field. An individual institution could then have their data re-ingested by GBIF or IHerbSpec in the same way that an institution might have its data re-ingested periodically, just as the data set is updated anyway. So this is a process that already exists and that could work. But again, the data would not be labeled as spectral data per se. You'd have to do a search into the notes section. So easy on the one hand, but not very operable on the other. The other option would be to take advantage of the Darwin core spectra or whatever the spectral data extension might be. And this would require the addition of a new field, for example, in IHerbSpec or GBIF. In IHerbSpec, it would be possible because we already went from a very small number, 15, 16, whatever we started out with, up to 82. But again, this would be challenging because we only have a year of funding left. So probably actually making this happen in IHerbSpec, while possible, would not be very likely. And I'm not sure how challenging this might be for GBIF in order to actually add another field. So, but actually getting the data in to those repositories would follow one of the same options as I mentioned before. Either for new specimens with new data, it would be straightforward. If you already have your records in one of these repositories, then it would require re-ingestion by the repository, again, following methods that are currently in use. But the last option is the one that I want to spend my last very few minutes talking about. And this is developing a new database that would be just for the spectral data. And this would involve in linking just the herbarium specimen identifier, that is a globally unique persistent identifier, and the spectral data. And that would be in whatever format is determined to be the best for these spectral data. And then the goal then would be to link the spectra with the complete herbarium record, which would already be present if that information were already incorporated into a repository. So again, I just want to emphasize the importance of having this be a globally unique persistent identifier for the specimen record. This is the type of record that one gets from, if your institution doesn't generate it, IHerbSpec generates it, GBIF generates it, the Symbiota portal system generates it. So there would be a unique identifier, maybe not the identifier that you use at a very local level, but something that's been assigned to meet these criteria. And then there could be multiple ways then to link the herbarium data and the records in GBIF with the spectral data in the database. And one possibility, and we tried this with GBIF data, so I've had a little experience with this, is that the staff at IHerbSpec could just search for matches and then flag the records in, for example, IHerbSpec as having spectral data. But of course, this would only be a short-term approach because this isn't something that could be done basically on the fly. This requires staff input on a recurring basis. But the other way, and this is something that you may all think is going to be crazy, but it could be to use a new approach called iChatBio. So I want to spend my last couple of minutes on introducing iChatBio. So this was recently released by iDigBio, and it was developed by my colleague Jose Fortes, who's a computer scientist, and his graduate student Michael Elliott, and the rest of the computer team that has established the structure for iDigBio. So you can get at this by going to iChatBio.org. Again, Jose and Michael are the architects of this. You need to register, which is basically add your email address and add a password. Then you log in. There's some background information, et cetera. So what is this? Well, this is the abstract describing this for the botany presentation that we're going to give later in the summer. And so, but that's way too much text. So I've just parsed this into some of the key points. So it's a biodiversity research-focused chatbot. I like to call it the bot, but it's not just for botany, it's for any biodiversity. It uses AI to answer users' questions, and it can assist with a whole range of topics, even now, and it's very quickly learning how to do all of this better. So it can help you find, retrieve, visualize, and process occurrence records. It's linked to data not only in iDigBio, but also GBIF and iNaturalist, and to the literature in the Biodiversity Heritage Library. It uses the same large language model as used by ChatGPT, and it also links up specialized workflows that can make all of this happen increasingly quickly and increasingly better. And then it uses a number of agents, and I'll describe that and show that to you in just a minute, to provide some other sorts of search capabilities. And then each of these new agents actually expands the capabilities and improves the performance. So this is sort of what it looks like. You could ask the chat agent a question, and that agent will pass on your question through an agent orchestrator. And let's say the first question has to do with data in iDigBio. The iDigBio agent queries the iDigBio API and then sends information back, and then that gets presented back to whoever has asked the question. And at the same time, so here are the links that we currently have in place, but it's also possible, because of this modular system, to add something else, and we'll come back to that in just a sec. So you can ask, like, how many occurrences of Acer rubrum does iDigBio have for Massachusetts? And you can get this answer back, and it says, according to iDigBio, there are 498 occurrence records, blah, blah, blah. And then it very politely asks you, would you like to see other information, such as collection dates or specimen images? So it can then sort of start a conversation and continue to give you more information. And so let's see. So then I asked which ones, I can't read that from there. Is this one about the images? Okay, so that's the beginning of it. And then I asked about images, and then it gives back an answer that you can click, and you get all of the specimen records that have images. And you click on any of these, and you get taken to the image that I showed you before for iDigBio. And you can continue to ask other sorts of questions, and I've just listed some of these here. We don't need to go through all of that. But I mentioned it's not just iDigBio specific. So what about in GBIF? Well, it does the same search for GBIF, but it got a little hung up here on trying to give me the information at the end, probably because I might not have been really specific about asking for just specimen-based things instead of maybe all observations, because it said it was like too much to report back. But it gave me options for further discussion. And if I wanted to ask something else that maybe makes use of another one of these partners, I didn't specify BHL at all. I said, show me the original species description. And it comes back and says that that's actually in species plantarum, so it doesn't have it there. Okay. So basically, where does spectral data come in? The spectral database that I described that could be established could be set up here as another partner. And you could ask the question, please return the spectral data for the species with occurrence records in iDigBio or GBIF. And hopefully, this would be the opportunity to bring the information from these two together. So how best to link the spectral data in herbarium specimen info. There are a lot of options, but I think that AI might help us think about how to approach this question in a way that's very different from how we've been thinking about extended specimens up to this point. So I'd just like to thank you all for your attention. I'm looking forward to the rest of the discussions, and I'd like to thank all my collaborators here who've contributed to this. So thank you.
Thank you. Thank you, Pat, for your presentation. That was great. And you brought a very important point, that is, what protocol we follow to upload this information to like a site such as GBIF or IDBio, right? So what one trend that it's being started at NASA with all our, like especially with the airborne missions, right? We do a lot of like validation in the ground. So we're also grabbing spectra in all those regions. So we're starting to follow the Darwin core, because we want to share that information as well in like GBIF and other platforms, right? So I think that's something that we need to keep in mind, especially thinking about your chatbots, because I'm sure if all the platforms follow a same sort of standards to have the information uploaded, it's going to be easier for the AI to cross-reference.
Definitely.
Right? And finally, I just wanted to put it out there, for every single like airborne mission that we have, so there is also a collection of like a dry spectra. So and that data is like publicly available, and I'm sure like that could also be useful for like work in herbarium, even though it's like most of the time is pulverized plant material.
Okay.
But it's still very useful.
Yeah. Great. Thanks a lot for your comments. I think that'll all come in really handy to the subgroup that's going to be working on that started working on the Darwin core. Yeah. Yeah. Anything else? Okay, let's go get coffee. Yeah, thank you.
doing great. Thanks. All good. You might have already pulled yours up. Yeah. All right, so you'll use this pointer if you want it, and then slides, and let me turn this on. Thank you. And then just
cool, right on time. Hi everybody, my name is Aaron Lee. I'm a 5th-year PhD candidate at the University of Minnesota with Ya Yang. And I'm super excited to talk to you guys today about some of my dissertation work on doing functional trait estimation and evolution from spectroscopy of herbarium specimens. Over the past 30-ish years, there's been this huge global effort to try and build to try and look into these global spectra of plant form and function. And these have sort of manifested into these huge trait databases, the most famous and widely used of which is the TRI database. But coming from a diversity perspective, there are a number of limitations with this database. One of which is taxonomic coverage of species that are actually included in the database and have traits measured for them. So on the left is an image of ecoregions across the world where in the TRI database there is a noticeable paucity of species representation across many diverse ecoregions. And Mason Heberling in 2021 made the case that we can actually use herbarium collections to try and fill gaps in these trait databases. So on the right is a plot from his paper showing that there's a huge amount of specimens that are basically untapped potential to try and measure the traits that are housed in herbarium collections. But there's this huge caveat that to measure functional traits, you actually have to be destructive, and that's really not feasible for herbarium specimens with limited tissue and limited material. And so our approach has really been to try and develop a non-destructive method using reflectance spectroscopy and start trying to chip into what's going on in herbarium specimens. And we're taking a lineage-specific approach to try and also constrain a little bit of what we're doing. And so in my case, I'm super interested in the family Polygonaceae. This is smartweeds and buckweeds and knotweeds. It's a family of about 1,200 species distributed all across the world. And what's interesting ecologically about this family is that there are repeated transitions globally out of tropical climates. And so this has a phylogenetic distribution. And so I'm not going to show you phylogeny here, but just trust me that there are 2 major subfamilies in this group. In the upper, the subfamily Polygonoideae, they originated in tropical Southeast Asia and dispersed all across the world into temperate and desert biomes. And then in the lower subfamily Eriogonoideae, they originated in tropical South America and dispersed northwards and then kind of back southwards across the New World into, again, temperate and desert biomes. And so in thinking about how to measure reflectance spectra from these species in this family, I just want to highlight a couple challenges that are things that we're encountering as we're moving through this in a lineage-specific way, and these are things that people have been talking about today. Oh, sorry. And then just to take a step back, I'm super interested in asking this question of how is climate and how is environment shaping the macroevolutionary trajectories of functional traits in this family. So now, okay, what are some of the challenges? There's huge variation in leaf size across this family, from huge, big leaves, some of leaves as big as a person that take up multiple sheets to the tiniest leaves to basically non-existent leaves. There is morphological variation between leaf surfaces. So from plants that have dense hairs on the lower side and basically sparse hairs like glabrous upper leaf surfaces to leaves where it's pretty much the same between the 2 surfaces. In this case, you've got stragose hairs on both sides. And And this question of like what is actually doing the function, this photosynthetic function in the plants. We have a number of species that are perennials and they have a nice sort of vegetative stage, and then when they shoot into their reproductive stage, they produce these big leafy bracts that are obviously doing photosynthesis as well and contributing to overall function of the plant. We also have plants where they've completely lost their leaves, and the photosynthetic function has transferred over to the stems, so we've got these photosynthetic stems, and it's also this really unique case of photosynthetic, or of C4 photosynthesis in stems. So how are we trying to measure spectra for, or I guess, so I guess once we've sort of decided how to measure spectra across this diversity, we also have to tackle the challenge of actually having traits. So this is a family that has good representation in something like the tri-traits database for things that are boreal and temperate, but pretty poor representation for things that are tropical, montane, and things in sort of generally in like South America, Africa, and Southeast Asia. And so what we did is develop a benchmark data set to try and build models from using species that we've, or samples that we've basically collected in the field here in North America, or in the USA. We've destructively, or we've prepared these specimens the same way that you would prepare a herbarium specimen, going through pressing, drying, and freezing. Did everything except mounting. And then we process these samples to measure some functional traits. Depending on sort of your evolutionary question or your ecological question, the traits that you want can vary. And we're trying to build, models that can maybe apply to as many situations as possible. But for some traits like leaf mass per area and carbon, nitrogen, and the isotopes, you don't really need that much tissue. But for traits like fiber fractions and mineral nutrients, you need a significant amount of tissue. So on the right over there is about 600 milligrams of Polygonum avicularis, or knotweed, a common knotweed, and that's multiple plants worth of leaves. It's really a significant amount, and like that's really not feasible to do with herbarium specimens in a general sense. And so with these traits and with these spectra, we're taking a partial least squares regression approach to build trait estimation models. You're going to hear a lot about these. I'll just give you a quick overview of like my understanding of how this works. It's dimension reduction. It's kind of like a PCA, where we take our spectra, we look at the variation that's present in our trait data, and we project the spectra to these latent variables or components that best explain the variation in our trait data. From that, we get these prediction coefficients that we can then apply to our data sets of interest, to our target spectra. And so we're using this sort of benchmark model to build a lineage-specific model from specimens that we've collected and can sort of vouch for their taxonomic identity and their traits, and apply that into the herbarium. So just to show you a little bit of what this benchmark model looks like, again, these are species from the United States, both temperate, subtropical climates, from the Midwest, the Western United States where the Rockies are, and the Sierras, and from subtropical Florida. It's about 44 species, and today I'm just going to show you results from leaf mass per area and nitrogen and carbon. You can see we've got good representation of most of the major genera in the family. We've got both subfamilies, and we've got pretty good coverage of the trait space in this family. Of course, from Flavia's talk, it seems like geography might be a problem in this model. So it's something to think about as we move forward with this. When we look at how these models do, we actually find that they perform reasonably well. You might be kind of alarmed. So we're looking at predicted on the x-axis, predicted traits, and then observed traits on the y-axis. We do a number of like model permutations and compare the models and try to understand what parameters are best in these models. And when we look at what the best models are doing, these R-squareds aren't like super high, but for our purposes, if we look at the error seems to be super low. And so this seems to be doing good enough for us to try and look at relative comparisons of how these traits are manifesting in specimens or in spectra. And so now with this benchmark model, we can look to what our target data set is. And so these are Polygonaceae spectra from herbarium specimens. I measured about 5,500 spectra from about 1,000 specimens covering 380 taxa. This data set spans from the mid-1800s to 2023 or 2024. And we've got pretty good coverage of North America as well as some diversity of specimens collected across other regions of the world. And most of these specimens are housed at the University of Minnesota Herbarium. I just kind of went through cabinet by cabinet, but also I was able to measure specimens from Wisconsin, Harvard, Herbaria, and from Missouri. And when we actually apply these models, we find that our predicted traits capture this distribution of functional variation. You see sort of with the colors, the different clades, and we see that as expected, that like there is sort of a conserved pattern of traits that are based on common ancestry. But my question is really about like what is the environment doing to shape the evolution of these traits. And one of the sort of key axes of environmental diversity in this family is this transition into more temperate, more seasonally variable environments or climates. And so when we plot out, for example, leaf mass per area against temperature seasonality, so the variation of temperature across a year, we see that when we do a phylogenetic regression using an OU model, that there is no relationship between seasonality and leaf mass per area. This is counterintuitive. This is not what we expected. When we sort of look at how LMA manifests into like plant strategies and the way that plants interact with their environment, we would expect a low LMA in a fast-growing and cheap leaf to do really well in an environment that's pretty homogeneous, where there are a lot of nutrients available, where they can kind of just do their thing and then die, move on, whatever. Whereas in a high LMA plant where it's tough and like stress resistant, where it's like super variable, we would expect them to do better. And so we were expecting more of a positive slope here. But when we think about some of the functional differences between these plants, and we pull out, for example, woody and herbaceous species and look at a phylogenetic regression between the interactions of this climatic trait, temperature seasonality, and woody herbaceous, we see that there is a difference. And so woody plants respond differently to temperature seasonality, and herbaceous plants do the same. And so in the context of the biology of this situation, we've got in herbaceous plants a range from fast growers and low LMA to stress tolerant slow growers and at high LMA and high temperature seasonalities, whereas in woody plants they just tend to be generally stress tolerant. Finally, I know I'm at time, I just want to bring it back to the spectra and think about how are spectra actually evolving, and thinking about this like spectra as a reflection of the functional context of these plants. And so what I'm showing here, I know it's like kind of complicated. I maybe don't have time, but if we look at the weights of these different evolutionary models, I'll be happy to talk about this more later.
Nice job, Aaron.
Good morning, everyone. I'm Charles Davis. I'm based right here at the Herbarium, and I am going to jump right in with the premise of my talk today, which is really about scaling science in a way that we really never previously imagined. And I would assert that the digitization of natural history collections and the online mobilization of biodiversity data more generally is really one of the greatest advances in biodiversity science since really the advent of DNA sequencing. And these data, of course, are fundamental to our efforts to model biodiversity. So we can look at this painting from Rachel Reich, and we can think about the species identities, we can think about the distributions, the kind of pattern, so to speak, and then we can go deeper and explore both the evolutionary and ecological underpinnings of that diversity, including the human ecological factors that brought these many species to Rachel's canvas in Western Europe in the 1700s, right? So the idea here is that these sort of digitized data really help us to build better predictive models to indeed make better inferences and choices about nature moving forward. And so since about 2007, we've seen explosive growth in online biodiversity data. This is a summary from GBIV. GBIV began very earnestly in 2001, and then, as you can see, grew dramatically at 2005. And on the right here, you can see that as these sort of online data similarly explode, so too do publications citing these data. So they are indeed stimulating tremendous and important science in our scholarly activities. The other thing I want to draw your attention to is this kind of more modest rise in digitization here. Those are the digitization of natural history collections, including herbaria, right? So much more low key than compared to the explosive rise in direct observational records. And this is a problem because we know from a whole variety of studies that these collections, natural history collections, capture tremendous taxonomic, geographic, and temporal diversity that are not represented in observational records. This is from a former postdoctoral affiliate of the lab, Barnabas Daru, and here you can basically see, you know, the differences in taxonomic, geographic, and temporal sampling in herbaria versus observational records. So they really are essential for filling out these essential dimensions of biodiversity. And so they're important for bridging this impasse, but the real challenge is most of these data remain dark for the world, and I call these silent collections, okay? So there are more than 400 million specimens that are distributed around the world. I should add they're distributed unevenly, and including about 3,000 herbaria, all right? And this is a challenge, of course, because there are all sorts of important things that we can and want to do with herbaria, all right? So the top three, the kind of what I call the bread and butter of herbaria, are taxonomic, monographic, and revisionary work, historical accounts of botanists and collections, and species distributions. These are just important today, arguably maybe even more important than they were hundreds of years ago when herbaria were founded, okay? And many people come on an annual basis to do just exactly these things. But over the last 100 years, we've seen a rise in other uses of these collections. So since the 1920s, we've seen growth in collections for global change biology, including things like phenology, for phytogeographical and range dynamics, for species distribution modeling work, and for biodiversity informatics, so simply growing, developing, generating the databases that we all come to rely on today. And so what I would argue is that we are undergoing a revolution in the use of herbaria. And this is really a result of a combination of 4 factors. The first is digitization of these collections. The other are large teams that bring in diverse expertise into these projects, the application of novel tools, and new domains of expertise. And this combination of factors is really propelling biodiversity exploration and discovery at an unprecedented scale. So this is a sort of conceptual paper that I co-led with Brandon Hedrick about 5 years ago, and what we did was sort of sketch out digitization in 2 parts, 1.0 and 2.0. So 1.0 is actually what's done in museums by museum staff dealing directly with the specimen. And usually what this means is capturing an image and transcribing a label minimally. And once these data are mobilized online, I should add here that there's a whole lot of road here to go in accomplishing this goal, 50 years, I would say, at least. And that involves just grinding it through, but also innovating new technologies like spectral imaging to get the job done, as it were. So 2.0 is basically utilizing the specimen, but only the digital derivative. So no longer working with the physical specimen. And the beauty of 2.0, right, is that you have these aggregated resources where you can do really science at scale. And so by that, I'm meaning here that we can do the science that we've always done with these collections, functional ecology, species distributions, morphometrics, adaptation, phenology, et cetera, but we can do it in a much more expansive global way. But the other thing we're seeing with these aggregated data is the influx of new domains of scholarship that are eager and excited to explore these data sets in a whole variety of ways, but also to collaborate with biodiversity scientists. And so those include artificial intelligence, as well as automated species identifications. This is a picture of a tour I have given dozens of times in the herbarium to students, faculty, even CEOs. And what you can see from this sort of table is the diversity of collection type that we steward, right? So you've got fossils, you've got woods, you've got slides, you've got alcohol specimens, and you've got even ethnobotanical things. This is a mortar and pestle to process chocolate. And digitizing these collection types requires a whole lot of thought and a whole lot of energy and a whole lot of exercise. So again, there's a long road to go on that front. Pam laid this out really nicely, the concept of the extended specimen, but we have, the physical specimen as well itself, and then you have these extensions, primary, secondary, tertiary, including things like a digital image, a DNA sequence, a tissue sample, an SDM, you name it. But the idea is that we're thinking about the specimen with this constellation of things moving around it, right? That enhance the specimen and really broaden the science that we can do with these collections. And so this is where the global metaherbarium really shines. It seamlessly accommodates this idea of the extended specimen. So I'll take a toy example here. We often collect in duplicate, all right? So let's imagine a collection event that has a specimen in Paris and here at Harvard and in Beijing. And in Paris, they've gathered morphometric data. At Harvard, they've gathered DNA data. And in Beijing, they've gathered secondary chemistry. So the beauty now is that we can interlink these specimens. That avoids repetition. We don't have to further damage a specimen to engage in some of these multi-omic exercises. And those data are available and accessible for the world to do the science that they want to do. It really is a nice accommodation of this extended specimen. And this is really important as we move into the future because of the whole variety of expanded applications that are ongoing and emerging in the collection sphere. So that includes next generation sequencing, machine learning approaches, metabolomic profiling, crowdsourcing for climate change investigations, mass spec analysis, isotopic assessments, spectral reflectance. You pick your your passion, all right? And I also want to reiterate something that Pam said, which is, it's just, it's like in my bones. Every time I give one of these tours, the first thing I think about is the possibility that are in these collections. I cannot tell you how many times I have thought about the fact that over the last 10, 15 years, and I've worked in a herbarium for 30 years, I would have never imagined the kind of work now I'm doing with these collections. So I think it's really important as we think forward to really keep in mind these future case studies and uses that we can no longer imagine, which become more and more important, especially as biodiversity is lost. And so this brings me to what I call the destructive sampling conundrum. This choice about making a decision to destroy a specimen. Let's be honest, this is the destruction of a specimen, at least in part, and the trade-off for the science, right? We could, if we utilize the global metaherbarium effectively and the extended specimen concept and linked data, making it public accessible, it's kind of a transfer of information that is still part of that specimen. But nevertheless, it's a destruction of a specimen. And this is just a little toy example from a paper that we published last year for kind of a DNA sort of analysis. And the important point is, we get lots of these requests for multi-omic investigations, and those requests are only growing dramatically, I would argue. And so this is just a simple plot of multi-omic requests granted from 4 institutions here at Harvard, the New York Botanical Garden, Missouri Botanical Garden, and Kew. But you can see that the number of requests have risen dramatically. And so, before I get to that, I will just say that like most of these institutions have guidelines for how to destructively sample a specimen. Our guidelines are not all similar, and our guidelines are not all equally detailed, okay? The other thing is that there's a lot of like anecdotal kind of information about best practices that floats around the community. And people that are 30 years in, like me, know and understand those kinds of things. But are people that are new to the field, they don't. And so we set about really putting our heads together across this sort of diversity of inputs and approaches to kind of outline a set of best practices for destructively sampling herbaria. And this is to 2 different communities that are partially overlapping. These are both users and stewards. And I'm not going to walk you through all these guidelines that we have, but rather you can read the paper, but I'm rather just going to kind of outline some key structural features of these guideposts. The first is that where possible, use other resources. Don't let the herbarium be your first line of sampling. The second is help us grow this collection, all right? That keeps it vibrant, all right? If you're going to use it, help us build it. The other is that always ask permission. You'd be surprised about the kind of stories some of us can tell about permission for sampling. The other is a set of like sort of develop your protocols efficiently and effectively not using precious materials, right? So only go after rare species, extinct species, unusual things once you've established the protocols. Annotate everything that you've sampled and make these data publicly available, okay? And lastly, and I really cannot stress this enough, we really need to, as a community, give better credit to the people that support and sustain and build research around these collections, and that should take place in the form of publications and grants. And I'm not talking about the one-line acknowledgement that is at the end of every paper. This is like obligatory, but I'm talking about, including people as co-authors on papers, including people in grant proposals that generate revenues for these institutions, because this is how they stay alive. We don't want to have any more situations like what we had just recently at Duke University. Okay, on the steward side, evaluate all sampling requests. I think it's valuable, we're still kind of working through this, but to treat multi-omic sampling as actually a part of the specimen. not an extension. It is part of that specimen. And so I think we can actually think about this more in the context of a loan, okay? We do need to protect against hoarding. I've got stories on that as well. And for those of us that have a career's worth of DNA sampling, for example, in our freezers, please make succession plans to accommodate this so that future generations can use those resources and we don't have to continually resample specimens. Ensure all permitting is up to date and on site, and then finally, make sure that these are accessible and transparent to all. Transparency is really important, I think, in this whole set of exercises. So, yes, this is a super exciting time. I couldn't be more excited about this. It's also a time for us to reflect and think about how to responsibly use these precious resources that are only going to grow vastly depending on the models of species loss that you believe as we go into the future. And finally, if you want to read more, these are the papers that I've drawn on for most of what I've presented here. So thank you so much. It's a really exciting group, and I think what's going on here is super, super cool and very important science.
Yeah, thanks. I just had one comment about these guidelines. At the end, maybe there was, I don't know if you alluded to that, but at one point it would be interesting to add a recommendation when possible to use tools or genetic data that's applicable to across outside of your study system. So in the Ananaceae project, we started off with very specific Ananaceae baits, but then Bill Baker from Kew said, why don't you just add in the 353 universal markers? And that was really an important part when we were trying to convince Herbaria for us to destructively sample, especially type specimens, only known from the type. Obviously, there was strong justification. And so now that the data is generated, we hope that nobody, I mean, if you want to use the 353 data, you can just go ahead and use the data that we generated. And when we can plug this into the bigger trees of life that are hopefully will be built throughout the year. So just as a comment to like an extra. I don't know if that was, I haven't read that paper in full.
We didn't touch on at that level of granularity. I think it's a reasonable point. You make one interesting point though, which we didn't all agree on in the co-author group, and neither do institutions, but sampling of type. So for example, we don't allow that here, but we also don't allow resampling. So generally, once you sample, you can't resample again. So it makes it all the more important that those data at some fundamental level be accessible, otherwise we change our policies.
Because it could be for both the users and the stewards, like, okay, you're going to do this, but make sure that the data is not only available, but it's also in some way extendable to other researchers, that you can just plug it in. So if somebody comes around, I'm going to sequence it for, this is phylogenetics, but I'm going to sequence it for very specific Ananaceae baits, then you could say, okay, you can do that, you can sample it, but make sure that you also try and sequence the other universal baits, which will make your study or the data more available across the...
Yeah.
And about the types, just a point, I mean, my project wouldn't have been possible at all, and we'd have really a very bad phylogeny if we weren't able to sample types, because many of these things are only known by the type. But obviously this, I was talking with Kimberly, is a lot of communication and discussion with the curators and explaining how this is going to enhance and be used. And you have to also come back to the collections once you've done this and say, okay, we've done this, and this is going to help you later on to fix some sort of taxonomy, or kind of come back to the origin and feed back some of the results to the herbaria that use it.
Yeah.
Yeah, thanks a lot, Chuck. I'm curious to learn your perspective on how does the argument towards the destructive sampling conundrum change given the current generation of devaluation of herbarium collections that did result in the closure of Duke, for instance? How does our sort of modern situation play into this? Does it change anything for you?
Look, I think we need to support cutting edge science. I think that is the one important way that we make ourselves viable institutionally. But that has got to translate to something. And the translations are in the papers we publish and the grants that we win that bring overhead revenue to these institutions. And I have seen any number of cases where individuals come in and gather hundreds to thousands of samples in a very short period of time, and win grants associated with that, that only very minorly bring value back to the institutions that are supporting and sustaining these. This is a big budget and a big footprint to keep this going, even at a place like Harvard, which is well endowed, you know, and well heeled. But I think we need to think more thoughtfully about how to better engage with these communities of people that help support these activities, that make sure these are safe for future generations of use.
So I just wanted to follow up on that. I just wondered to what extent there's a risk with the metaherbarium and the sort of global herbarium that actually that might do the opposite, in that once the data's available, that there's a lot for like AI mining and lots of stuff around sort of property. Once they have the data, people don't need the herbarium to exist anymore, right? I mean, is there a risk that you're kind of factoring in with that?
I can't tell you the number of administrators at a variety of places that said, once we digitize it, can we put it away?
Right. Yeah.
And I think that's just, anyone who's worked with a collection can tell you, you need collections for a whole variety of functions that an image or a spectral signature or even a whole genome sequence can't provide. And so, you know, having that level of accessibility facilitates repeatability in the science that we do and allows for the expansive work that this workshop is here to actually focus on. So I think it's really short-sighted to sort of imagine, well, once we've got it all imaged, can we box it up and put it away? Because what I would argue actually is that digitization is doing quite the opposite, which is it's bringing more and more people here on site to see, to view, to study our collections, and to think about new ways to explore them with new realms of science.
I hope you're right, yeah.
Thank you, Chuck. That was brilliant. So along the same lines, I wonder, you know, what your thoughts are in how to keep herbaria vibrant, right? To how to keep people seeing them as dynamic places, or actually incorporating new specimens and looking out in the world, figuring out what gaps are out there and targeting things. So I guess one of the worries is, there are millions of specimens out there, and people just think that that's enough, and you don't have to keep collecting. So
I just got a message that I should keep this short, but I'll say that it is essential for the vibrancy and resiliency of any kind of natural history collection to continue growing and building that collection, and keeping pace with exciting science. I there is no bigger champion on this planet for herbaria than I am. I mean, some may be equal, but I will proselytize as much as I can about the exciting science that's going on. Sadly, much of it is not, it's not recognized, it's not brought forward that these were actually derived from herbarium specimen data. And that happens from all different kind of domains that are using these resources. So for all of you that use them, please do highlight that this is where they come from, but yeah. I saw one more question, okay.
Okay, thank you very much to all the speakers this morning. We're now going to transition into the lightning talk format. So we can see here we have 12 people. We're going to try and keep this to about an hour with 5 minutes each. So we're just going to rotate through and then have some time again at the end for questions. So forgive us for not quite having enough space to have questions with each talk, but we think this is a better way to do it. Okay, so we're going to start off with Natalie Iwaniski Alstrand from the Natural History Denmark. And I guess I will just briefly introduce the people since I know who's coming next, but we'll just come up and move right through them. Thanks. Natalie, please come up. Yeah, it's all on one slide. Yeah.
Switching slides.
Switching slides. So we can use the clicker to move forward, or the arrows. And again, we need to try and speak.
Right. Thank you, Dawson, and thank you, Jeannine, and everybody who has made it possible to gather here together. I'm coming from Denmark, from the Natural History Museum, but I'm from Montreal originally, about a 5-hour drive away. And I worked actually in herbarium curation and in plant conservation in Toronto area for many years before I landed at the Natural History Museum. And I've been there for 11 years now. And only in the last year have I started in a new position, a tenure track position, where I'm also a curator of digital botanical collections. And myself and a couple of my colleagues at the museum have interest in really pursuing the hyperspectral I guess, research field and world, and I'll get into a little bit of detail, but we don't yet have any spectral equipment. So we've been, for the last 3 years, trying hard to get funding and infrastructure up in place to match or mirror our digitization efforts, which I will talk to you about. It's been really exciting from a North American perspective and the history that we have here with collections to move to an institution with an even longer and very impressive history. Our museum, actually, the Natural History Museum, is an amalgamation of several independent museums, zoology, botany, and geology, which were brought together about 20 years ago. And we're in the process of building a brand new natural history museum, which is actually slated to open in early 2027. So I hope that I can see many of you coming to visit us there. We have in total about 14 million specimens and about 3 million in terms of our botanical specimens. There's been a renewed focus. It's also been kind of an eye-opener leaving North America and the scene and the pace of digitization that has been happening here for the last 20 years or so, and moving to Europe and thinking, oh my goodness, so many collections, and nothing has happened until all very recently. So embarking on this sort of, maybe second, third, fourth generation of digitization pipelines has been pretty excited. And our new museum will have a museomics lab and a digitization lab as well, where we expect a lot of these new methods to come together. We are a part of the DISCO initiative in Europe to digitize scientific collections. We run at our museum, we're the leader for the Danish system of scientific collections, and that's led by my colleague Kim. And Kim is a computer scientist who's half appointed at our museum, and his interest is in hyperspectral imaging, which I'll get to in a second. So not just the scanning, but full images. We have financing for the next couple years. It was a 5-year-long process to initiate our digitization workflows, and we have to self-finance the coming years. So we're sort of at a crux right now of figuring out where to go from there. But we have really good infrastructure in place already for plants and for entomology collections, and we have some really exciting projects working on new technologies and robotics to handle 3D objects and scanning, and including incredibly large collections from whale and dinosaur bones and skeletons to all the way down to microscopic material on slides. We have been reliant on Specify, like many of you here, and are publishing not only to some of the well-known biodiversity platforms, but also our own Danish biodiversity platform, which houses from citizen science and crowdsourcing types of data to our collections data and any observations of biodiversity for the country. In addition to that, or as part of that workflow, something that we're really are capitalizing on right now for the next phase of our infrastructure development is the transcription end of the process, where we really will rely on the modern workflows today of AI and machine learning to help us with that process. However, we really want to connect people to that process. And again, what's next? Well, how do we get in any kind of hyperspectral scanning or imaging into the process? As I mentioned, Kim, my colleague, he's been using hyperspectral imaging, where an image of an entire object is taken. In the case that he's worked on most is with the protein content of wheat grain, and we've pitched many times already to try to apply this. It hasn't been done yet for dried plant specimens like Herbaria tissue. So you could take an image of the entire leaf and you get thousands of images representing all of the spectra. And then computer scientists are really needed to mine that data. So our plans are to, or have been, and we will continue to do that, to try to raise funds for infrastructure and PhD and postdocs that will continue down with both point-based spectral scans, but also the whole imaging on a number of plant groups. Some of them very tricky, the ones that I work on, graminoids being very thin-leaved, and or lycopods, very small leaves that can present challenges. But I'll leave that there, and next is Olwen.
Thank you, Natalie. Next is Olwen. So this is Olwen Grace from the Royal Botanic Gardens, Edinburgh, and Mike Hopkins, you're next. All right.
Morning, everyone. Good to see you. So my introduction this morning is just to tell you a little bit about who we are as a herbarium in Edinburgh. I am Deputy Director of Science and responsible for our preserved collections at the Royal Botanic Garden, Edinburgh. Our herbarium code is E. As you will know, having a single letter as a herbarium code in Index Herbariorum is indicative of, we like to think of our stature and status as well as our age. So like many places in Europe, but not so much in the UK, the origin of the Royal Botanic Gardens, Edinburgh, was very much as to support teaching at the University of Edinburgh in the 17th century. And so we really have our roots in that sort of physic garden history, which is more typical of European gardens. The herbarium itself was really formalized about 150 years ago, although our collections do date back to the 17th century. The Royal Botanic Garden, the Royal Botanic Garden Edinburgh actually encompasses 4 sites, including the Edinburgh one, and there's some factoids there about how diverse our living collections are. And in addition to our preserved collections, so library, archive, including our art collection, and the herbarium, we also have an extensive living collection which is angled very much around biodiversity conservation and increasingly restoration and rewilding in Scotland. We also have a thriving educational program. So we have a relatively famous master's in taxonomy and biodiversity, but we also have a very vibrant adult education and schools education program as well. The herbarium, we think, has about 3 million specimens, but we won't know until we complete our digitization. The image on my first slide was our celebration of reaching the milestone of 1/3 of the herbarium digitized, so imaged and databased. And for that, we held an outreach event with a secondary school's holiday camp. So the students came in to help with the digitization of the millionth specimen, which is really good fun. We are the national collection for Scotland. We're the other national collection the other national collections in the UK are obviously located in Wales and at the Natural History Museum in London. We continue to be extremely active in accessioning about 30,000 specimens a year. These are either collected by our own researchers, or they are gifts that are coming increasingly from a generation of very active citizen scientists in the UK who've built up their own private herbaria. We're about to embark on a further scaled up digitization. So we currently have 6 full-time digitizers. We're going to be scaling that up as part of the DISCO UK. So this is the sort of European equivalent of iDigBio. And we will be working closely with the National Museums of Scotland to provide a national hub for digitizing natural history collections as part of that program. We are fortunate to be supported by a very engaged sort of public-facing team at the Royal Botanic Garden. And so actually currently, if you happen to be in Scotland, we've got a lovely exhibition up featuring the herbarium in our visitor experience building at the John Hope Gateway. But what I will do in the last 4 seconds, I've got 30 seconds. What I wanted to highlight was our research interests. So like most other herbaria, we support really large, intensive programs of collecting for sequencing. But I'm also leading on research to integrate automated identification and AI, specifically into curation, but also around species identification. We're about to procure our first instrument for measuring spectra, and the intention is to dive right in with some really tricky groups, including the ferns and succulent aloes. So we're jumping in at the deep end. Thank you.
Thank you very much, Olwen. Next, we have Mike Hopkins, from the herbarium curator from the INPA Herbarium.
Yes, we're a 4-letter herbarium, so we're we're really quite recent. We were founded only in 1954. We're 70 years old now. We're in Manaus. Manaus is a huge metropolis in the middle of the Amazon. Used to be well forested. It's most famous for the meeting of the waters of the Rio Negro with the Solimões, the Opera House, and of course the Flora of Reserva Duque, which I have as a PDF if anybody wants it. Unfortunately, it's not available to buy these days. So we are the herbarium there. I am the curator. This is it. Okay. Amazon, it's been spoken a little bit. We have a massive biodiversity, probably totally very, very poorly estimated. Those numbers that Flavia was talking about, 16,000. Personally, I think that's probably a half or a third of the actual number of species that are present in the Amazon. Massive area in which to work, and unfortunately very low investment in the most fundamental studies. Quite a lot of investment in climate studies, etc. these days, and in inventories, but very little in taxonomy. INPA Herbarium, there we go, 300,000 collections, small by North American standards, but pretty big in Brazil. But large investment in inventory plots. Unfortunately, in Amazonian forests, the trees do not flower all the time. They do not flower every year. So the vast number of making collections in the inventories, which I really stimulate people to do, unfortunately, they're nearly all sterile. And we really, our eyes opened up with the studies of Flavia of the possibility of near resolving the problem of identification in inventory plots. What material do we have? We have the herbarium, have a duplicate collection, which I'll be talking about, and we have the inventory material. Problems, here's the decline in the number of technicians contracted in the herbarium over the last 15 years. If you take that line there, that's the reciprocal of the time that the curator has to do work in the herbarium there, and well, no, so it's the line also for how much time I have available to invest in new projects, to invest in looking for new sources of funding, etc., because I'm basically doing curatorial work all the time, doing technical work all the time. Problem. Lack of funds, lack of flexibility in use of funds as well in Brazil. I have no actual status. I have no financial resources I can call on. Any financial help that I get, it has to be through an application to me to some kind of funding agency. Nothing by the day. This is a modelling that I did some years ago about the it goes a bit more there about our knowledge of the Amazon, which I say is very little. That's those areas there circled are the ones where we have least information about the taxonomic composition of the plants. Here's a distribution of our collection, no, not our collections, of collections in the Amazon on a large database, and you see concentrated in particular places, near cities mostly. And that one over there is an estimate of how far behind Amazonia, the green graph is, compared with the other parts of Brazil, which I haven't have time to talk about, but it gave an estimate of a minimum of 65 years that we are behind the rest of Brazil in knowing the flora of Brazilian Amazon. I can talk about that. And here we are, Amazonia, sorry about the Portuguese, more than half the area of Brazil, but with less than 9% of the taxonomists there, and actually much less now. Those arrows are all taxonomic botanists or mycologists who have left the Amazon area over the last 10 or 15 years. So that number of 9% is quite a lot less now. Hopeful signs. We do have some hopeful signs. 1 minute. The NEAR, we've used it successfully, as Flavia was saying, in various student projects. That we are going to get 2 new taxonomists, one mycologist and one higher plant taxonomist there. We recently bought 22 NEAR scanners that Flavia talked about. We have a very well, modesty aside, we have a very well organized database in Brahms, which is a very good platform for working this. And we recently received funding for a new herbarium, which will be in that building there, 100 meters long, 15 meters, 10 meters tall, which will be a vastly improve our space for working, which we have none of at all at the moment. 20 seconds. And this is an idea of where we will be able to do scanning. We actually are in a good position because we have a lot of duplicates that have never been mounted. Because of the lack of technicians, I'm also about 5 years behind in our mounting. So I have a lot of material that's waiting to be mounted that would be excellent for scanning. If only I had people to do the scanning, because that's the problem. But as you can see, basically most of the arrows there are coming from material that is still unmounted. And also a lot of our mounted material is mounted with thread rather than glued. So it will be better for scanning. So we're in a good position to do a good project. Oops, I'm over time. And just in Brahms, this is a sort of model of what we will do in Brahms. I hope we will have an interface to facilitate the scanning process and automatically labeling the files as we bring them in. And it's a very good program. We will be able to link up with all other databases, I hope. 30 seconds over time, sorry. Thank you.
Barbara. Yes. Okay. Barbara Netto-Bradley from Cambridge University.
And I'm going to be talking about trying to identify species in the genus Lithocarpus. So this is a group of tropical stone oaks. And I'm coming into the picture really motivated by using these specimens to address a series of different research questions. But ultimately, in order to do that, those specimens need to have a species name attached to them for most cases. And when I was looking through the specimens of Lithocarpus, I realized that there's this growing knowledge gap, which I think many of you will be familiar with in other lineages and in other places. So this is just a plot of the proportion of specimens that have been identified to species versus, are sitting in a in-depth folder for the genus. Towards the present, there's this uptick, and it's not sort of a 10 or a 20-year lag, and someone's still going to come along and have a look at it. That really starts in like the 1950s, and I don't think that gap is going anywhere unless we try and address it. So for lithocarpus, this is often the case because the key features for identifying them are mature fruits, and there are so many specimens that are just vegetative. So for this, spectra seems like an ideal potential way of adding value to these specimens that we might not be able to identify otherwise. And so I'm going to tell you a bit about using reflectance spectra to do that, but I'm presenting it alongside using a computer vision approach as well, so that you can sort of see how it might do relative to some of the other automated methods that we've got. To do this work, I headed down to Kew, and other people have mentioned the difference between the leaf and the leaf on the page. And I'll just say that I only use the leaf measured on the page for this because, unfortunately, almost all of these specimens are thoroughly glued to the paper. And I think there can be other conversations about how we might detach these as the day goes on. So I trained, well, with the help of folk from PlantNet, we trained 2 different versions using data from the same set of specimens. So we've got the spectral data on one hand, and then the whole RGB image on the other hand. And just sort of along the way, in terms of the spectral approach, I was interested in thinking about how many specimens we're going to have to look at per species in order to do this. This number is going to vary depending on what taxa you're looking at, sort of a whole set of different things for lithocarpus and for this set of species. Within that, we found that there is this breakpoint at around 30 specimens. Again, that's going to vary, but we went forward with things that we had at least 30 specimens for. And then I was interested in thinking about how many species we could differentiate in one go. And you might want to think about this if you have additional data sets that you could use to sort of filter or select what to feed into the spectral model. There's a steady decline, but yeah. So back to the 2 different ways of doing this. On the left, we've got the spectral classification, and on the right, there's the PlantNet version. This is the confusion matrix, but actually, I think an easier way to visualize this is the overlap in where both models agree in their predictions. And so for 61 of the 102 test or validation specimens, both models agreed with each other, and that was the same as what was on the herbarium label. But what I think is really interesting is that each model was able to identify things correctly that the other model missed. And also, both models were able to identify things, or co-identify things that were different from the herbarium label. So there's, I think, some neat opportunities for, 1, using spectra alongside other data sets, and 2, for sort of using these tools to flag specimens that might otherwise fly under the radar, but could be sitting in the wrong folders or have interesting stories attached. So with that, I'd just like to acknowledge the help that I got and the conversations I've had along the way. I don't know who is Matt, you're next.
Thank you. Yes, we have Matt Austin next from Missouri Botanical Garden, and following him, Samantha Bazan.
Okay. Hi, everyone. Thank you for having me and all of us. I think this is just great to bring everyone together. So I'd like to tell you about some of our preliminary work on trying to automate identification of North American vitis at the Missouri Botanical Garden. So vitis, the grapevine family, this is obviously a very important group economically. Their fruits are used for, grape consumption and fermentation. They're also an indigenously important group. Vitis material has been used by indigenous cultures for food, medicine, toys, and ceremonies. And we're lucky to have a relatively well-curated vitis collection at Mobot. However, there they're a taxonomically tricky group, and this is partially because polymorphic leaves make identification difficult. So we're interested in trying to automate identification of this group using hyperspectral data. So the preliminary work that we've done so far is using a NaturSpec spectroradiometer, full range with a 3 millimeter spot size to collect data on 11 Vitis species, and we included one Rhicinodendron species in our model as a type of outgroup or control. Every species included 10 specimens, and we took 3 abaxial and 3 adaxial measurements per specimen. Collection dates range from the late 1800s through early 2000s, and we avoided specimens with glue and pathogens and used a non-reflective black background for each measurement. We then trained partial least square discriminant analysis models, PLSDA, using the first and second derivatives of the hyperspectral data combined, and used a 60-40 split where for each species, 6 specimens were used for training and the other 4 for validation. And we iterated the PLSDA 30 times with random assignment of specimens to training and validation in each iteration. And we found relatively good accuracy. Across iterations of the model, we achieved about a 90% accuracy. And so the numbers in tiles here are percentages. So you'll note that many species have accuracies between 88 to 100%. But I'd like to draw your attention to 2 kind of right in the middle, Vitis riparia and Vitis estivalis, which respectively have a 67% and 74% accuracy. And I was curious to try to dig into why those 2 species have a relatively lower accuracy than the others. So to do this, I made a plot where on the x-axis you have specimen, and the y you have the percent of times the specimen was incorrectly predicted. So every line is a specimen, and all the way on the right are those that were incorrectly predicted a lot. And notably, the majority of specimens that were incorrectly predicted more than 40% of the time are those 2 species, Vitis riparia and Vitis estivalis. So I said, okay, this would be interesting to pull those specimens and give them to our in-house Vitis specialist, Aaron Floden, and let's see what he keys them out as. And notably, there were 10 Vitis specimens that were incorrectly predicted 40% or more of the time, and 4 out of those 10 he keyed out as being incorrectly determined. So here you have the confusion matrix for those 10 specimens. On the right, you have what Aaron keyed them out to. And so notably, he keyed them out as different species than the confusion matrix is suggesting. But 4 out of the 10 were incorrectly determined. 2 out of the 10 had insufficient characters for correct determination, for keying out, and 4 of them were determined correctly. So I mentioned this just to highlight. It seems obvious, but even when specimens have a species level ID, they won't always be correct, and it's something we should be mindful when using these data to automate species identification. So thank you.
Thank you, Matt. Next we have Samantha Bazan from the CIRAD Herbarium.
Thank you. I'm sorry, I'm not so fluent as the other speaker, so I will keep my notes. So I'm the curator of the CIRAD Herbarium in Montpellier, France, and today I would like to present NIRS as a potential tool for herbarium curators to improve the quality of identification in their collection. So I have the feeling I'm talking about herbaria in front of an engaged audience, so I will be brief on this point, but herbaria are truly remarkable and irreplaceable sources of information on biodiversity, millions of specimens available. But in many herbaria, we have some issue of identification. So many unidentified specimen by lack of time, expertise, or sterile specimen, as we already talked. Name changes over time, of course, and new species are described. And we also have some misidentified specimen, maybe not in the Harvard Herbarium, but at least in mine, and I think I'm not the only one. So can NIRS could help us to improve the quality of identification in Herbaria. To test this hypothesis, we used a case study with model genus Pistachia with 5 closely related species. We scanned 160 herbarium specimens from 11 countries, collected from the 18th century until last year. On this material, we took 7,000 spectra on the adaxial side of the leaves using a pen-shaped probe and a black reflectance paper. We used an SD portable spectrometer and took measurements directly in a collection, as you can see in the picture, in Paris Museum. We start in an exploratory way to describe the spectral response of 4 main species of Pistachia. We observe a relatively clear structure of the 4 species, which are well discriminated. Oh, it works. Great. Sorry. During this descriptive analysis, we visually spotted some suspect points. As you can see, they were clearly misplaced in the modeling. For this suspect specimen, we checked the image online and sent it to specialists for verification, and all were confirmed to be misidentified specimens. So thanks to NIRS, we can find and correct identification for those old specimens in Herbaria. And it's an interesting potential for a curator like me. In the second step, we also want to test the taxonomic prediction on newly collected specimens from a training set of old herbarium specimens for the 5 species this time. We run a PLSDA, which gives a success rate of 87%. It's promising, but our dataset is light on some species. So in the future, we need to increase the number of specimens to consolidate it. So the first results show that NIRS allow to detect and correct some misidentified herbarium specimen in collection. And this method seems promising to help for the identification of unidentified specimen. But we need to perform analysis on more data on different taxa from different institutions, different ages, to confirm the efficiency. And I'm sorry for this, we have already seen this at a previous meeting, but we decided to share our data set of Pistachia spectra and data. So you can find and download all the data by scanning the code or just asking me. You can choose to play with a raw file, or if you're not used to deal with spectral data, we also offer a preprocessed file ready to use. Thank you very much.
Thank you, Samantha. Next is Etienne Levey-Boré from the University of Montreal, and following him will be Khalil.
Hi everyone, so I'm curator of the Marie-Victorin Herbarium at the University of Montreal, and I'm also a plant systematist, like a lot of you. And so one of the main uses of plant spectra that I'm interested in is using them to identify species and potentially other uses related to plant systematics, evolution, phylogeny. And one of the first questions I had when I started to learn about plant spectra and this kind of uses was, what are their limits for plant identification? So what I decided to do is to use the worst possible group to test the limits of plant spectra, which is hawthorns. Hawthorns are a notoriously complex group taxonomically because there are about 400 taxa, and they might be varieties, they might be species, they might be microspecies. We don't know for sure because most of them are actually tetraploid, probably allopolyploid, and there is very little genetic data on these species. So I wanted to know whether we can differentiate hawthorn taxa, I will call them genotypes, based on spectral data. And I wanted also to know how we can deal with fuzzy classes, which means that some of these individuals that we're measuring might be hybrids, so they might not fit exactly in one of the classes. I want to know also how can we recognize taxa that are outside the training set, because a lot of these samples that we might be feeding into our analyses might not have been included in the training set at first. And I also wanted, and that's related to everything else, to be able to predict genetic distances between individuals based on the spectral data. So what I did is I selected a relatively limited easy case for that, around 20 taxa, genotypes. I won't use the term species here, in Quebec, so a single province of Canada. I collected data on 400 pressed specimens, so that's a relatively easy to analyze spectral data set. And we measured 3 leaves per specimen on both adaxial and abaxial side, the reflectance, a single spectrum per side. I should probably have collected more, and probably I will in the future. So this is all the other thing that we used, if it's of interest to you. One thing that we noticed is that there is a lot of unexplained brightness variation within a specimen, and that's probably due to microtopographical differences when the leaf is not perfectly flat, so specular reflection issues. So if you have a way to correct for that, please let me know. That's really complicated. We also collected genetic data using RADseq. So the first thing we tested is just the classical approach, PLSDA. We saw a lot of that in the previous presentations, and we will keep seeing that. And so what we found out is that we can predict fairly accurately a lot of the classes that we identified using genetic data. So we did not use just what was on the label that was not related at all to what the genetic data were saying to us. And we can predict very well these classes. The thing is that the classes that we use in this model, because it's based on genetic data, they're not all species. So you have series, sometimes a species, sometimes a variety or potential hybrids. So we have different taxonomic levels, and it's a bit difficult due to this taxonomic complexity of the genus. So I wanted to test another approach. Instead of classifying into classes, I wanted to predict genetic distance between specimens, and so I used a PLSR approach, which we use for traits normally. So what we did is we compute genetic distance based on the RADseq data. With this genetic distance, we project using an ordination. And then what we use is we use the PLSR to predict where a new specimen will fit inside this ordination. So I'll just go really quickly. It worked quite well. So this is the predicted genetic distance. This is the real genetic distance, and this is the one-on-one line. So there is a good correlation, but it's not perfectly linear. So this is our current problem. We're testing new approaches. If this is of interest to you, or if you have ideas to fix that, please let me know. I'm really interested to know more. And that's it. Thank you.
Next we have Khalil Bughalmi from University of Grenoble-Alpes, and following him will be Reichardt Jackson.
Thank you, Dawson. Yes, it's better. So I will just present you the first step we done with Thomas, because Thomas already showed you an overview of the project I'm involved in. And the purpose of the project is obviously to do some more evolutive analysis, but to do that we first need to see if we can have a species signal.
Try to use the microphone.
Sorry.
No, it's better like that. So yes, the purpose of what I'm going to present you is to see if we have a species signal in the spectrum data in Herbarium. So as Thomas said, our main goal is to analyze the diversity and the evolution of tropical forest using Annonaceae species from Herbarium specimen. For doing that, we built very big model, and so we need very big data sets. We have already morphological and genetic data sets, but we want to try to use a new kind of data, the spectral data. The main interest for us in the spectral data, as Thomas said, is that they, like we expected, represent the chemical and the structure of leaves, and so we assume that this particularity are linked to like the evolutive history and the environmental pressure on the species lives. We only use Herbarium specimen because we want to have as much as possible species sample, and we have the chance, as Thomas presented you, to have very good quantity of specimen in the analysis. But as I told you, we have one main question: if like we can use those kind of data for doing analysis at the species level, because what we can expect with spectrum data from Herbarium is that we have like a huge time span, but also like a state of the leaves who are very different from one and other. For doing that, we built two different data sets. A first one in the Paris Herbarium, where we selected 12 species with as much as possible specimen, and we only selected the specimen who was recently revised in a monography. And we also try as much as possible, when, like, if it's possible we do that, but sometimes it's not possible, to take the most widely geographic origin for the specimen. So we end with this data set with 25 different countries for the spectrum we have. We also want to test if there is a difference between like an old specimen and a new specimen. So we take like very old specimen for some cases, and we have a time span of more than 190 years old. Another question we have is more like related to the preservation method, but also the drying method and this impact on the specimen spectrum. Because, like for example, in our cases in a tropical forest, one widely used method is preservation in alcohol, and alcohol is widely known as a powerful solvent and oxidizer for like just a chemical compound, and so we was afraid of this impact on the spectrum. And the point is that in an herbarium we don't know what is the kind of preservation. We generally don't know what is the kind of preservation that the specimen has been through. We, for both of the data sets, just like for the testing data of the preservation method that was built in Yasuni National Park by Thomas and other colleague he has, and so we have identified the specimen who has been preserved in alcohol. For all of the specimen we have, we use an ISD lab spec. We take 20 points per specimen only using the adaxial surfaces, and then after that we standardize the data, and we use for all of our analyses the first derivative of Savitsky-Golay. As Etienne said, we also find that a lot of people using the spectrum specimen use a PLSDA, but we want to try also like a SVM, support vector machine, who can be very effective with the high-class analyzers. So we have, for the Paris data sets, a very good result with more than 90% of good accuracy with all the different specimens I told you. So we were very surprised. It's a good result. Both with SVM and PLSDA, who had like slightly the same kind of result. Just so you know, with SVM, with the analyses are more than 25 faster than PLSDA. For the Yasuni datasets, so as I said, we try to see if the alcohol have an impact, and surprisingly, there is no impact, or like not a big impact, on the species discrimination using alcohol specimen. And that was something we are very happy with that, because we can now start to scan more species and not like considering the fact that can be like alcohol specimen. So we are very happy with the fact that we have like the capacity of identifying species, discriminating species from Herbarium specimen, and now, as Thomas told you, we will start to use this kind of data for modeling more the evolution of tropical forests.
Thank you for your attention.
Thank you, Khalil. Yeah, so now we'll be transitioning into some talks on traits, starting with Rykar Jackson from University of British Columbia.
Cool. Yeah, hi everyone. Yeah, so my name is Rykar Jackson. I am a master's student at the University of British Columbia in Vancouver, Canada. My presentation today is going to be a little bit of a follow-up from the presentation that I gave back in March. So I'm sorry if you're just joining us for the first time, this might be a little bit out of context, but hopefully I can string something together that makes sense. But since then, in that presentation, I just talked about some of my preliminary findings, and since then, I've been able to do some of my first analyses. So I'm happy to share some of my first findings with you all today. So broadly, my project is looking to quantify the effects of spectral degradation over time using this experiment utilizing ginkgo biloba, and we have put those specimens into these 4 different temperature treatments to simulate degradation. They increase in severity, so we can kind of monitor how that increase in degradation impacts them. Now, 2 years into the project, we have 3 different sets of data. So we have the fresh leaf spectra, the dry leaf spectra after 1 year, and then the dry leaf spectra after 2 years. It's also worth mentioning, too, that we have 2 complete sets of samples, one from the early part of the season and then one from the late part, but that's kind of outside of the scope of what I'm going to be talking about today. Excuse me. If you guys might remember from the presentation in March, one of the main takeaways that we had was that we actually saw the highest amount of variability being in the herbarium control, which was really surprising. We've kind of chalked it up to it being an effect of the facility in which it was stored not being properly temperature controlled, which I think speaks to the importance of temperature control in herbaria, but I'm kind of preaching to the choir, so I'll just leave that at that. But in such a short presentation today, I'm just going to focus on how this finding was reflected in the PLSR models that we just completed. So with that being said, if the slide will switch. Okay, there we go. So yes, God. Okay. I'm gonna assume it's gonna go back. Jeez. Great. Okay. My apologies. So this is just the broadest model that we created. So it just took all of the dried leaf spectra, separated it into 75% training and then 25% testing data. The only manipulation that we did was that we stratified the selection by treatment just so that each one is represented in each side. So you can kind of just see, we are only looking at these 3 different traits. They're the area-based traits, so that's leaf mass per area, leaf dry matter content, and equivalent water thickness. The chemistry-based traits are just currently held up upstream, and I'm hoping to get my hands on them soon. But just for today, I'm just going to talk about those 3. All in all, we can say that the models perform moderately well. Going back to what I was saying about that variable herbarium control, we can see that that gray line associated with the herbarium control is the most askew from the diagonal. So that makes sense considering that those were the most variable going in. So the models should have the most difficulty predicting those. You might be looking at the R-squared values and saying like those are decently low. And they are kind of low in the grand scheme of things, but when you look at similar studies using pressed leaf spectra, they do kind of lie within the range of variability. I do also think that they might have been a little bit deflated due to some inaccurate area measurements. We already took out the extreme outliers that were obviously mistakes, but there are still a few, I guess I'll call them suspicious readings that require reanalysis, but unfortunately those are also held up at that same place with the chemical data. So once I get my hands on those, it'll be really interesting to see how or if the models change. It's also worth noting that in relation to each other, LMA and EWT are better predicted than LDMC, which aligns with previous findings, so that is quite promising for me. But with that all being said, if the slide will change, God willing, I'm going to assume it's going to change. Sorry?
The arrows.
Yeah.
The
Yeah, that one's also, no, there we go. Okay, sweet. So what I was just going to say, this is just the exact same model, just with that variable herbarium control taken out. So we can see that with the removal of that source of variability, the error does consistently go down, which makes sense. But the R squared does change in either direction, which is kind of strange, but I'm interested to dig into that next. That's kind of my next plan. But I will just end by saying that with these values, the R squared values specifically, we do kind of see there's like some sort of relationship happening here, but it's the same question that Etienne has, where it's just like, there's something there, I'm not sure exactly what it is. So if one of you thought that and you're like, oh, I'm going to tell Etienne what happened and I know, tie me in as well. I'm interested to hear it. So all in all, I think based on these preliminary results, we can say that after 2 years in the degradation treatments, it appears that there isn't really a significant impact on them. We would see that red line be the most askew consistently, and that's not what's happening. So I'm hopeful looking forward. And that is all. Thank you all.
All right, thank you, Raikar. Next we have Sean Kotari from the University of Alberta.
Hey everyone, I'm Sean. I kind of made a fatal mistake in that I kind of, in my mind, I had it that that this was like an ESA inspired talk where it's like auto events every 15 seconds. So I have 20 slides. So this will be a bit of a whirlwind. But you know, if the worst case scenario is that I get a hug from Jeannine, that's not the worst thing. So I think it's really cool that I think it's really cool just to reflect on the fact that these ideas about measuring spectra on herbarium specimens just kind of originated independently multiple times. You know, I first talked about this with Dudu, I think in like 2016. I was like, do you think this would be possible? And you and Jeannine, you mentioned that you and Jeannine had already talked about it. Yeah, right. Yeah, you know, most ideas don't pan out, but this one did. And like, you know, so I didn't know about the work that, you know, like Flavia Durgante was talking about just now, and others have been doing elsewhere. Natalia is probably thinking of the same things. So I just think that's cool. But we had the opportunity to pursue this when Jeannine and I talked with Etienne Laliberté here at a meeting of a research coordination network. He mentioned that his student Rosalie Beauchamp-Rioux had collected some hundreds of samples and you know, measured the spectra of the fresh leaves and had also measured traits and collected herbarium, or not herbarium specimens, but like pressed leaves to eventually be, some of them at least, accessioned into the herbarium. So we had like 600 or so samples to work with, and a collection of all these traits which conveniently had already been measured. And then we measured, so the fresh leaves had already been measured, fresh spectra, measured pressed leaves, and then there were ground samples that had been used in chemical analysis. There's extra, and we were able to measure more. And you know, so you can see some examples of the different spectra, including different different specimens, including different conditions, including discoloration here on this poplar, poplar there. And yeah, so we had the chance to test out how well does trait prediction work. You can see for a trait like LMA, you know, we kind of predicted that this would be something that would work best from the fresh leaf spectra, and from ground leaf spectra, there's not really a reason for us to be able to predict it directly, because you know, once you grind up a leaf, the LMA is sort of meaningless. It still kind of worked, which you know, we can talk about why. But with the pressed leaf spectra, we were kind of pleased to see that, like, yeah, it still pretty much works. We were able to predict it, and I think the results of many other people today have also kind of reflected that. You know, LMA is a trait that we can predict pretty readily. And then there were other traits that went the other way. The fresh leaf spectra were the most, you know, had the worst accuracy, and going towards ground leaf spectra was more accurate, kind of reinforcing what Jeannine had mentioned. One cool thing, you know, inspired by what Matt and what Samantha was saying about how, you know, this inspired people to go back and look at original identifications, is that for the model for nitrogen, which is also like this, like kind of like a straight line, there was one point that was like way out here, like kind of sticking out. And I went to Etienne's lab manager, Sabrina Demers-Thibault, asked her like, can you reanalyze this specimen for nitrogen? And she did, and it fell right on the line. So it was just nice to see, like okay, sometimes these spectra are more sometimes they can inspire us to go back and look at the actual, what we think of as the ground truth data. And we also are able to compare fresh and pressed leaf spectra for species identification. This is not like a particularly difficult problem. These are not closely related species. There's a matter of convenience. But I think the cool thing is the ability to compare. And we saw that pressed leaf spectra and ground leaf spectra as well did better for the species identification task than fresh leaf spectra. Next, I want to talk about another project that we launched more recently. This is Juan Posada at Universidad del Rosario in Colombia. And he was visiting Quebec, and he was like, he was interested in kind of seeing, could we predict photosynthetic traits? I will leave it to you to decide if this is a good idea or not. I'm not going to make any claims there, but we were like, how far can we push this? And so we had more than 100 samples from around the Montreal area. We had some like funky things like these rhododendrons with fuzzy undersides of the leaves. And we measured various photosynthetic traits as well as spectra. And for the fresh leaf spectra, we only had visible and near infrared. So for something like saturating photosynthesis, like light saturated photosynthesis, we can see that we do fairly similarly with the fresh leaf spectra and the fairly recently pressed leaf spectra, not herbarium, but it's fairly recent. And for something like electron transport rate, maximal electron transport rate, also we end up doing pretty well. Of course, like there's a lot of reasons why, maybe people should not be doing this, but I think this reflects something about sort of what we can capture about the underlying bases of these traits, things like the nitrogen content, et cetera, that help to determine these physiological rates, and maybe there's something useful there. So yeah, this is all stuff that I don't need to talk about. So yeah, yeah, cool.
Okay, thank you, Sean. My name is Dawson White. I'm a postdoc here at the Harvard University Herbaria, working both with Chuck and Jeannine. And as Jeannine mentioned first thing this morning, we here are sort of extending what Sean just presented about looking at these pressed leaves that were collected in his part mostly in southern Ontario, and saying, well, we have this study system, we have these traits that were measured on these species. What if we go into the herbarium and now measure those same species? I should say it was a subset from 68 down to 25 here. But let's measure those same species and specifically look at one trait, LMA, and then perhaps see what else is going on with those other models for the other traits that Sean generated. So this project is it's going to be published fairly soon. It's on EcoEvo Archive, and Jeannine was part of it, Chuck is part of it, Antonio Guzmán, Sean Dudu, who's here, and Jorge Robles, who is an REU student this summer at California State University, San Marcos. So the first thing I want to discuss here, present, is what really is the difference between herbarium and pressed leaves, okay? So pressed leaves, by themselves, these are generally in newspaper. These are things that have been collected in the field. They've been dried. All of us are making these pressed leaf collections before they're then deposited in the herbarium. And yet, when they get to the herbarium, then they're mounted, and we are then dealing with an agglomeration within an herbarium collection of all of the different collection methods, all of the different protocols for processing the leaves, drying them, sterilizing them, and then ending up with mounting, right? And yet many herbaria across the world, their collections are still within newspaper or newsprint. Okay, so they're not always going to be mounted across the globe. But we are dealing with ages that are different. So up top here, this is the actual ages at the time of scanning from Sean and colleagues' study, and versus 1 to 179 years. Those were the ages from the Harvard University Herbaria specimens. That just exemplifies a large difference with much longer ages expected in herbarium leaves and typically mounted on archival sheets versus loose, and a large variability, a difference in the variability we can expect across these specimens, okay? But overall, perhaps we should be thinking about pressed leaves really as a subset of the variation that we're seeing within the herbaria. All right, and with all of that variation, we can assume that we are going to encounter much larger conditions of leaves, and that is going to translate to variability in spectra. So in moving forward, we were kind of speculative as to how this could work, but here is the validation data showing LMA collected from the herbarium leaves by itself, generating a model from the herbarium leaves, and we could see that it actually worked quite well. So this is a wonderful result. Here the colors are by species instead of by growth forms that Sean showed. And sorry, this is the press leaf spectra. The herbarium spectra then, this is what we saw, okay? The accuracy of these models was still very good. This is a great result, positive outlook. Then we wanted to say, look more deeply at what are the real differences that we're seeing here. On the top, you can see in black the mean value and the 90% quantile of the spectra across all the species from the herbaria versus some higher reflectance in the pressed mean spectra. But within the models that we generated, we can see these variable importance of projection, the wavelengths that were really presumed to be important in these models. They really overlap along the x-axis in the places that are inferred to have higher importance within the models. So that's good. That's showing us that the models that were built with pressed versus herbarium look fairly similar. And then the same thing, the model coefficients on the bottom, it gets a little messier, but that's showing us these nuts and bolts of how these models are working. When we compare them, they're actually more similar than expected. And even more so when we apply this transformation, which Antonio has sort of leveraged and taken, which is continuous wavelet transformation. So this is sort of similar to a derivative approach. And then if we use, furthermore, how comparable are these methods? Well, if we take our herbarium model and apply it to the pressed spectra, which is on the right, we can see that the herbarium, I'm sorry, the other way around. So the herbarium model applied to the pressed spectra, we're still getting pretty decent accuracy in one model being applied to another one, and then vice versa on the right. So a side note here then, one of these species is clearly out of the range of the rest of them, and this is Agonis flexuosa, which is actually Australian, the rest of them being North American species. And what I want to show you really quickly here is that Agonis has very high LMA, so it's extending our trait space. And if we remove it, well, our models actually don't work quite as well. So we were seeing, some along the higher values of the models that were shown earlier, how the models sort of start to fail. Well, that can be as to how you actually built your training model that, was seeing this amount of variation. So we need to be considering both the taxa that are going in as well as the trait variation that are going in. And along the lines of taxa, here quickly looking at some of the other traits that were modeled, calcium and cellulose, we're seeing, and again, we had no validation data here for the herbarium. These were just models from pressed applied to these other traits. Generally, nothing was negative. First takeaway, these look sort of okay. Some traits better than others, as expected, and some taxa performing worse than others. In this case, it's two grasses, Phragmites and Phalaris. Okay. And that's it. I do have a summary slide. Yeah. Thank you.
So thank you, Dawson. So hello, everyone. My name is Antonio Guzmán. I'm a research scientist here at Harvard University working with Jeannine on integration of ecology and remote sensing. I don't know nothing about herbaria. I know nothing about specimens, but you can talk me about spectra. And so what I'm going to show you today is actually, it's not results, it's actually a vision about how we can use spectroscopy, how we can do integration in the community to better understand what is biodiversity. And so, okay, this is not my slide. Okay, this is mine. Okay. And so, as Professor Davis laid out, the utilization of specimens are kind of driving the new discovery and use of herbarium specimens. And this new area where we have spectroscopy, of course, that provides a new venue to understand of biodiversity and doing phenometric biodiversity information from specimens. But as we advance or we start the collection of data, we may be wondering what we can do with that, how we can share that, how we can curate that. And the difference with an image that you can scan from a specimen, usually spectroscopy data that comes from the leaf, requires certain kind of processing, requires certain kind of harmonization between spectrometers, between kind of methods that were used. And so we need to think about how we can use that in order to leverage future efforts. If you have data, we may consider to put that in a platform. And there are really some platforms out there in the spectroscopy community. You can see, for instance, this one, the Ecological Spectral Information System, EcoSys, or you can go to this other one. It's called Open Soil Spectra that just focus on soils. And so perhaps there are platforms out there where you can put spectra data, but this platform not necessarily have all the needs that we need to consider in order to work with the specimen information. And so what we are kind of proposing in this vision is to develop a platform that helps us to support the exploration, the visualization, and analysis of spectroscopy data derived from herbarium collections. And so how we have been calling this, we have been calling this Earthsphere, Herbarium Spectral Hub for Research and Exploration. And the idea is to have a platform that can support what is exploration and the engine of deriving traits from spectroscopy from herbarium. This was a proposal that we submitted to Harvard Data Science Initiative. We don't know if it's going to be funded given the current uncertainty, but of course the idea is to support what is the dissemination information from H to H. Of course we are thinking big, as because this may be funded, maybe this is going to be sources using APIs. Of course, we can perhaps integrate other herbaria with the help of all of you. And so what this project could do? So what we can envision, with this? Well, we've tried to see what is available, what we can see at a geographic location, what we can see for institutions, what we can see with taxonomical spectral information. And the main goal of this is to do querying exploration, how we can get information on that. And one good example of that is the Open Soil Spectral Library. So we don't need to invent the wheel. We can use the really information that's over there and adapt that to our needs. And this is an example that you can go to, for instance, Costa Rica, select one point, and see all the spectra that they have for that location. And this is just for soils. And there are different kinds of sensors that they use. But the most important, you can go down, and then you can pretty much select what is the area of interest and download the spectral information for that area of interest. And so this can complement the study of biodiversity for that specific region. And it's not just one institution, it's a lot of institutions out there. And so this could be an example for that. And then we had other element, which is the engine, how we can predict traits from that. And the prediction of traits is no easy task. You have a lot of requirements, a lot of thinking about how we can improve the models, how we can do these models with bias. And so the goal is to have a way that we can analyze, predict, and assess traits derived from these spectroscopy information from specimens. And one example of this could be, for instance, our current app that is called Spectrates, where you can get spectra, you can envelope that information, and then you can have coefficients for a given model that were created that you can then use to validate and predict traits. You have your own data sets of traits. For instance, you can envelope them. You can select a trait of your interest, and you can potentially validate those, how well those are, and download that information in order that you have a good documentation of that. And so with these slides, my goal here in these presentations is outline this kind of vision that we need as a community, a way that where we can share information, we can build better models, and we can potentially do a better study of biodiversity. And with that, thank you so much.
Great, thank you, Antonio.
Sorry for being before your lunch.
So we're at 12:20. Yeah, it's time for a photo at this point. Should we?
What about our panel discussion?
Yeah, what about the panel discussion? Should we just hold off or go into lunch?
How about let's do 10 minutes of panel discussion, and then we'll do our photograph, and then we'll have 15 minutes for lunch.
Okay, great. So we got 10 minutes of questions for lightning talk speakers, or
Yeah,
all
just lightning
speakers.
talk speakers. How about you all sit up front?
That's good.
And well, 10 minutes of questions. And, the people can. I'll
Should we just-
moderate the questions unless you want to do it.
Should we just, no, you go ahead. Should we just stand?
Okay.
We've been
Yeah, just
sitting.
stand. For 10 minutes, you can do it. So, if you just gave a lightning talk, this is our chance to ask you questions. I'm going to pass around the microphone. Is there anybody left to ask a question? You can also ask questions of each other. So.
I'll ask a question.
All right, go for it.
All right, so I had one for Khalil. You mentioned that part of the filtering that you did was for specimens that were actually cited within monographs. Is that true?
Yes.
Yeah.
Yes.
What was that process like for the number of specimens that you had? How efficient it was, and versus the actual value that was gained towards your confidence in that taxonomic assignment?
Maybe one thing I can say is that that was the first time in my life I was in the herbarium. So I think it's maybe a good representation of someone who want to do that kind of thing, but don't know exactly how we need to use herbarium specimen. So Thomas gave me all of the monography of the genus we selected. We previously selected the genus who are the most widely distributed in some area, for example, Africa. It's a Paris herbarium, so we have a lot of data from Africa. So we selected the most widely distributed and the one with the most specimen in Paris. And we, I just take the box of the specimen of the species, and I find the specimen I wanted. I use 2 days, I think, just to sort all of the specimen. I will, after that, scan. So yes, approximately 2 days for put out close to 200 specimen, approximately. But that was time consuming, I can say that. But for some cases, when I find a specimen who's been labeled as recently determinate, I think it's that way, but with very strange looking, I take it also as a supplementary and scanning them to see how it works with discrimination method. Yes.
Thank you.
Hi, thank you all for amazing talks. This is more just generally so anyone can answer, or maybe some popcorning. Spectrometers are not cheap, and I know some people touched on trying to apply for grants and get instruments even and just work with them. I guess, does anyone have ideas on how to make that more accessible, or if there can be sharing between institutions? Barbara?
I'll go first, I guess, because for my study, I actually borrowed a spectrometer from, in the UK, there's a facility that is, I guess, sort of equivalent to your NSF-funded body, and they maintain the spectrometers and also train people on how to use them. And so I think something like that could be an option for broader access, but I don't know what other people have to say about it.
Barbara, you can keep the mic. So, I mean, your work is fascinating, and I'm wondering if you could elaborate a little bit on the potential of unifying the two disparate models that you have in a way that you can leverage, image recognition, traditional morphometrics, right, with spectroscopy in a way to, so the models can play off of each other's, like complement each other.
Yeah, thank you. So actually when I went through this, I didn't design it as I'll do both at the same time. I was thinking only about the spectra on the first pass. And the comparison came up later in the project, so it wasn't designed from the get-go as let's use them together, and that's why it was kept separate. But I think the next step naturally is to use both models together. And I tested one approach, but I didn't have that many specimens left for training and doing that. So I think, yeah, there's a couple of different ways you could integrate the different sources of information, and I hope someone else tries them. We're just talking this morning about, the folk at PlantNet, I think, are interested in this too.
Yeah, cool. Thanks everybody for really awesome talks. For those of you working on species identification, a lot of you are finding, okay, the specimen that you're identifying is actually misidentified. So has anybody tested what goes on if you include a misidentified specimen in your training models? And then kind of second part to that question is has anybody approached it from a species delimitation perspective and done more unsupervised learning approaches to try and separate between species using spectra?
Well, I have not tried including misidentified specimens in the model, and I think the questions you're asking are the very reason why I think we should think about moving outside of just classification and into other approaches that really integrate the genetic similarity between specimens and can predict the similarity or the position in the phylogeny, or think about other approaches to that problem, because classification algorithms will only work if your training data is good and if classes are clearly delimited. So.
Could I add to that? So I can clarify for the Vitus work that we've been doing, so we were able to flag some specimens that were misidentified, and those were included in the model because we iterated the model, the PLSDA, multiple times with random assignment of specimens to training and validation. Those misidentified specimens were sometimes in the training data and sometimes in the validation data. So I mean, the effect seems to be that it caused the accuracies for those species to be lower. But again, by iterating the model multiple times, where they were also included in the validation set, we were able to flag them as potentially misidentified. We haven't yet rerun the model with, specimens that should all be correctly determined. But yeah, it lowered accuracy as you would expect.
Yeah, we saw that in the.
Let's do one more question.
Sorry.
Oh, go ahead.
Can I
Well,
just?
you and then
We saw that in one of the papers that Flavia mentioned in the Burseraceae, that with a revision of the identifications and redoing the models, the accuracy of identification, predicted identification, increased immensely, which is always my hope that this method would actually give a good reason to invest in taxonomy, to make taxonomy great again, as somebody was saying yesterday, because we will need it. We will need good taxonomy to do good models for identification.
All right.
Our last question.
Great presentations all. My question is to Reka. In the models you presented, you had your trait values, and then you had R square, and then the performance levels for all the different traits. Was the performance based on the average of the different conditions, because you had different treatments? So was it for the general? Was it an average value from the different conditions you presented per trait? And then the second question is, how did you, you spoke briefly about phenology, you had early stage, and then you have late stage, like senescence. And can you talk a little about it, and how did you tear apart or separate the different leaf stages?
Yeah, so the first question is an easy answer, is it was unaveraged. So that's that. But in terms of phenology, we ran all the same models with like early and late lumped together, and then early by itself, and then late by itself. And we didn't find that it, especially just for such a short presentation, early and late didn't differ so much that it was worth talking about. But when it was all lumped together, it kind of gave a, it was almost, no, it's not even more, it's not almost, it was a better model in the same way that Dawson talked about how when you have a larger trait space, the models have more to work with. So we found that when they were lumped together, that variation actually did help.
Okay, thank you, everybody. The sun has come out. We're going to do a photo outside.